So good morning dear students. From today onwards we will be starting with the special senses. Actually this is a continuation of central nervous system, as we could not cover the special senses portion along with the central nervous system. So we will be starting from today. So in special senses we are going to learn about the physiology of vision, the physiology of hearing, the physiology of taste, smell and the equilibrium. So why these sensations are called the special sensations? Okay, in CNS you have learned that the sensations, they are broadly classified into the general sensations and the special sensations. So the general sensations, these receptors, they are not very specific for a particular type of sensation, isn't it? Whereas if you take the special sensations, you have some specific type of receptors which are hosted or which are placed in a specific sensory organ, and that is the reason these sensations are called the special sensations. For example, if you take vision, we have some special receptors called the photoreceptors. Then for the hearing process, we have the auditory receptors. So likewise, we have some specific receptors for the different types of special sensations. Okay, so the scheme for today's class goes like this. So before discussing about today's topic, I have put some of the MCQs. Totally I have put 5 MCQs. Just take a paper and mark down the answer for this. You need not have to post in the group. Okay, this is just to check yourself how much you have understood at the end of this class. Okay, I'll just read the question. When light passes through the lens system of the eye, it first encounters, that means when the light is falling on the eye, the first layer of the retina which comes in contact with the light is this the layer of rods and cones? Is it the pigmented epithelium or the ganglion cell layer or the inner limiting membrane? So please note the answer for this. The next question, the photoreceptors present in fovea centralis. Okay, the photoreceptors present in fovea centralis are the rods only, the cones only, or is it both the rods and cones or none of the above? That means whether it is not having any type of receptors. So you have to mark it and tick. So coming to the third question, this is bit tricky because you need to align these answers in an order. Okay, so the structures forming the refractive media of the eye from anterior to posterior aspect are, the structures forming the refractive media of the eye from anterior to posterior aspect are, the tear film is the A, B is the aqueous humor, then C is the cornea, D is the vitreous humor and E is the lens. So these are the various refractive media which are present in the human eye. Now you have to arrange these refractive media in an order. So you have to mark the answers. Is it the combination of A, B, C, D, E or A, C, B, E, D like that? So you have to mark the answer for this. Now the fourth question, the normal intraocular pressure of the eye is about, is it 12 to 20 millimeters of mercury, 30 to 40 millimeters, 42 to 50 millimeters of mercury, or is it 52 to 60 millimeters of mercury? Then the last MCQ, aqueous humor, it is secreted by which of these cells? Is it the corneal epithelial cells, pigmented epithelial cells of the choroid, or the epithelial cells of the ciliary process of ciliary body, or is it by both cornea and the choroid cells? Okay, so please mark the answers and keep it with you. Then towards the end you can check by yourself. So coming to the scheme for today's class. So in today's class we will be learning in detail about the functional anatomy of the eyeball, and also you have to explain the physiological importance of the aqueous humor. So let us quickly start with the topic. So coming to the eyeball. So eyeball is a very important structure, isn't it? So today if we are seeing the external wall, that means we all are blessed with a normal functioning eye, isn't it? If we don't have a normal functioning eye, we wouldn't have appreciated the details of the external wall. So this eyeball, it is a spherical in shape. Usually they tell it is like a globe. Okay, so the eyeball is like a globe, and this globe is flattened from above downwards. Okay, this is flattened from above downwards having the antero-posterior diameter of about 2.4 centimeters, approximately an inch. Okay, 2.4 centimeter antero-posterior diameter, and having a vertical diameter of about 2.35 centimeters. Okay, so where is it present? So this globe is been placed in a bony structure which is a pyramidal shaped structure called the orbit. So the eyeball is placed in the bony orbit, which is deficient. This bony structure is deficient anteriorly, and within this bony structure the eyeball is suspended by means of extraocular muscles and the facial sheaths. Okay, probably you would have seen in your anatomy dissection class how the structure is present in the orbit. Now coming to the eyelids, if you normally you have this normally this eyeball is protected by the two structures, that is the eyelids. Okay, you have the upper eyelid and you have the lower eyelid. Okay, and the space between these two eyelids, they are separated by a space called the palpebral fissure. So this eyeball is having the medial, the angle of the eye, and the lateral angle of the eye. Towards the medial angle of the eye, you can see some raised mucus or the raised tissue. So this is called the caruncle. So this caruncle, why it is important means it has some sweat glands and the sebaceous glands in it, which helps in maintaining the lubrication of the eye. It keeps the eye in a moist conduction, and because of the presence of the eyelids, so whenever the wind is coming very fast or something, even the dust particles which have some microbes or any foreign bodies, if you close your eyes with the help of these eyelids, then it will be prevented from entering into the eyes. Okay, so that was about the eyelids. Next comes the conjunctiva. You all are familiar with this term conjunctiva. In the general physical examination also we have shown there are two types of conjunctiva. Conjunctiva, it is nothing but the mucous membrane which overlies the inner aspect of the eyelids and the white portion of the eye. So the portion of the mucous membrane which lines the inner aspect of the conjunctiva, that is the palpebral conjunctiva. Okay, palpebral conjunctiva, wherein you are going to look for the presence of pallor and other signs. Next, bulbar conjunctiva is the portion of the mucous membrane which overlies the white portion of the eyeball. Okay, so that is about the conjunctiva. And one more important aspect of conjunctiva is, as you all are aware, this conjunctiva, since it is in close contact with the external environment, it is more prone for infection. The inflammation of the conjunctiva is called conjunctivitis, which is usually characterized by the redness of the eye, the irritation of the eye, which will make the person to rub his eyes more often. Okay, the best example for this conjunctivitis is the Madras eye or the red eye. Coming to the extraocular muscles, as I was telling, the eyeball, it is placed in the bony orbit which is suspended by means of some muscles. These muscles are called the extraocular muscles. There are 6 extraocular muscles which include 4 recti and 2 obliqui. So the 4 recti are the medial rectus, the lateral, this is the medial rectus which is responsible for the medial turning of the eyeball, then the lateral rectus for the lateral movement, then the superior rectus for the upward movement, and inferior rectus for the downward movement. So along with this 4 recti, it is also provided with the 2 other important muscles called the superior oblique and the inferior oblique muscle. They are together called obliqui. The 4 rectus muscles are called recti. Okay, that is the plural form. Now coming to the innervation of these extraocular muscles. These are mainly supplied by 3 important cranial nerves. So how will you remember? So this can be remembered as LR6, SO4 thrice. LR6 stands for the lateral rectus, which is mainly innervated by the 6th cranial nerve, that is the abducent nerve. SO4, that is the superior oblique, which is mainly innervated by the 4th cranial nerve, which is nothing but the trochlear nerve. And the thrice means the remaining all the muscles of the eyeball, that is the extraocular muscles, they are supplied by the 3rd cranial nerve, that is the oculomotor nerve. So this is again one more diagram showing the different muscles. So this is the eyeball, which is surrounded by various extraocular muscles like the medial rectus, which is present towards the medial side, lateral rectus, inferior rectus, superior rectus, and here comes the oblique. The inferior oblique, this is mainly responsible for the downward movement, and superior oblique, which is mainly responsible for the upward movement. Now coming to one more important structure which is present in the eyeball is the lacrimal apparatus. So most of you are aware the eyes are continuously covered with a thin watery fluid which is nothing but the tear. So this tear is formed in the lacrimal apparatus. So where is it located? This lacrimal apparatus is located in the upper lateral portion of the eyeball. Okay, so this gland, there is a presence of lacrimal gland which goes on secreting the watery fluid called the tear film, and this tear film, once it is secreted, it will reach the lacrimal sac via the lacrimal canaliculus. Okay, through the canaliculus, you cannot see here. So through the canaliculus it will reach the lacrimal sac, and from the lacrimal sac the tear will go into the nasal cavity via the nasolacrimal duct. Okay, so whenever there is blockage in the nasolacrimal duct, so what will happen? So there is obstruction to the flow of the tear, that is the water which is being secreted by the lacrimal apparatus, and it will get collected in the eyeball itself and the eyes will start watering. Okay, so that is the importance of this nasolacrimal duct. So tear film is nothing but it is a fluid covering the cornea and the conjunctiva. So this is very important in order to keep the eyeball in a moist condition. Next, it is mainly made up of the tear. It mainly contains 98% of water and 1.5% of sodium chloride. As it contains sodium chloride, what could be the taste of the tear? So it will be saltish. Okay, so it is salty in taste. Now, what are the functions of this tear? It contains various antibacterial substances which mainly try to prevent the microorganisms which are being entering into the eye, and thereby it will provide the immunity to the eyeball. It minimizes the infection of the eyeball. So antibacterial substances like lysozyme, beta-lysine, lactoferrin, they are present in the tear film, and it also has the mucus which again is necessary for the lubricating function, and the antibodies also they are present in the tear film which is mainly contributing for the immune response. So that was about eyeball. Now coming to the layers of the eyeball. The layers of the eyeball are called the tunics. So which are the layers of the eyeball? The eyeball is mainly surrounded by three important coats. These are called the tunics. Okay, tunic is nothing but the layer or the coat. The outermost coat of the eyeball is formed by the cornea and the sclera. The middle coat, it is mainly formed by the choroid, ciliary body and the iris, and the inner coat, it is mainly formed by the retina. So concentrate on this diagram. This is a simple diagram of the eyeball. So the outermost layer of the eyeball, okay, especially the anterior most portion of the eye, so this is formed by the cornea. Next, posteriorly the cornea continues as the sclera. Okay, so this is the outermost coat of the eyeball. Next, the middle layer, this outermost coat, the outermost coat is called the fibrous layer because this is mainly made up of fibrous tissue, and this in turn consists of cornea and the sclera. The middle coat, it is highly vascular, so this mainly consists of choroid, ciliary body and the iris. So if you concentrate here, so this portion, here you have the ciliary body, okay, here you have the ciliary body, and next you have the iris, the pigmented structure. Here it is the ciliary body, then the iris and the choroid. Okay, choroid is again a vascular structure which is present all around. Okay, so the middle coat, it is mainly consists of the ciliary body, the iris and the choroid. Okay, next the inner coat, it is mainly having a neural component, and this is mainly formed by the retina. Okay, so the innermost layer of the eyeball, it is the retina. Okay, the reddish thing you cannot make. If you can, you just concentrate, and if you observe carefully, the innermost reddish thing is the retina. So these are the three coverings of the eyeball or the tunics of the eyeball. Now coming to the outer coat. As I told, this outer coat, it is mainly formed by anteriorly the cornea and posteriorly the sclera. So the anterior 1/6 of the eyeball, of the outer coat, it is very transparent. Okay, it is very transparent, and this is called the cornea, whereas posterior 5/6 it is very hard, tough and opaque. Okay, so this is opaque, it will not allow the light to pass in. So this is called the sclera. The junction of these two is called the limbus. The corneoscleral junction is called the limbus. This central, the cornea, it is nothing but the most central and the anterior portion of the eyeball, and this is highly transparent. The light rays can just pass through the cornea in order to get focused onto the retina. So this allows for the light to pass through, and one more important property of this cornea is it has got a self-regenerating property. If there is any damage, it can get repaired by itself to some extent. Now, other features of cornea are it has got very less number of pain receptors. Okay, it has got a low pain threshold, but it is highly sensitive to touch. Okay, the cornea, it is highly sensitive to touch. Suppose if you bring your hand in front of you, automatically you get a blinking response, isn't it? And it is highly avascular. One more important property of cornea is avascular. This is contributing for the transparency of the cornea and for the entering of the light rays into the retina. So this is also contributing for the process of image formation. Okay, and it is not having any lymphatic drainage. And then since it is avascular, how does it get the nutrition? So the cornea gets its nutrition in the form of glucose, which is available from the aqueous humor. Aqueous humor, which is a fluid which is present in the anterior chamber of the eyeball, which you will be learning in the further slides. Next, the gaseous exchange, the oxygen and CO2, they get exchanged directly with the external environment, that is the atmosphere itself. And the refractive index of the cornea, it is about 1.376 anteriorly and 1.336 posteriorly. So the refractive index of the cornea, it is about 1.334 on an average. So you would have learned, you would have observed this cornea in your histology classes, and you have studied that it is made up of 5 layers. I am not going in detail of the structure of the cornea. So that is corneal epithelium, Bowman's layer, substantia propria, Descemet's membrane, and the corneal endothelium. Now coming to the applied aspects. So usually what will happen, the cornea, as the age advances, there is a condition called the arcus senilis. Arcus senilis is nothing but, senile means something to do with the aging phenomenon. Arcus means it is an arc-like structure. So arcus senilis, it is usually seen in old individuals. There will be a formation of peripheral opaque ring around the cornea, and this is seen with the age. So why this ring is formed with the age? So this is because of deposition of some of the lipids like the phospholipids and the cholesterol. So next is the corneal ulcer. Since the cornea is the most exposed area of the eyeball, if there is any damage to the epithelium, the corneal epithelium, it can lead to ulcer formation, or it can also because of trauma. Okay, and these corneal ulcers, if they are present, they will interfere with the process of vision. Next, the corneal graft, as you all are aware, eye donation is nothing but we are not taking the entire eyeball and we are going to transplant it into a new individual, isn't it? It is just the cornea we are going to extract from its donor, that too especially within 6 to 12 hours immediately after death, and it should be transplanted within certain time. Only then the vision will be gained by the recipient, and this is the most widely accepted the transplant, because cornea is a avascular structure and there is no fear of graft rejection because of avascularity. Okay, so that is the clinical importance of corneal graft. Now coming to the sclera. So that was the anterior 1/6 which is mainly formed by the cornea. Now the remaining 5/6 portion, it is mainly formed by the sclera. So this is a fibrous protective layer having both elastic tissue and the fibroblast, and sclera is nothing but the white portion of the eye is called the sclera. And this sclera, it is thinnest at the limbus. Limbus marks the sclerocorneal junction, and it is about, here it is about 0.7 millimeter thick, and it is thickest at the posteriorly by about 1 millimeter. There is one important structure which is present at the sclerocorneal junction, which is very important for the flow of aqueous humor, and this is called the angle of Schlemm or the canal of Schlemm. Okay, so this is present at the sclerocorneal junction. So this is just to show the cornea, the most anterior portion, followed by the sclera, posterior 5/6. So then you have the middle coat. Now let's see. The middle coat of the eyeball, it is mainly called uveal tract because it is mainly comprising of the choroid, the ciliary body and the iris. So what is this choroid? The choroid, it is a blood-rich nutritude tunic or the layer, and there is a thin pigmentation which is present in the choroid, which is very much needed for the prevention of the scattering of the light. So when the light rays are coming from a far object or from an object, so what will happen when it passes through the cornea? So there is a chance for the light rays to get scattered. So this is prevented by the thin pigmented layer, that is the choroid. And this choroid supplies, being highly vascular, it mainly supplies the inner retinal sensory cells with the nutrients and oxygen. This is very important because if there is any damage, if the innermost layer gets separated from this layer, what will happen? The retina will be deprived of nutrients and it will be deprived of the blood flow, isn't it? So that condition can permanently damage the vision. So this choroid, it supplies the inner retinal sensory cells with the nutrients and oxygen. That means the retina is also depending on some other structure for its nutrition. Next, the movement of nutrients into the sensory cells, it is mainly done by the active transport mechanism across the cell membrane. So the important point to note here is the vision deteriorates when the retina is detached from the choroid. That is because the nutrition of the retina, it is dependent on the choroid. So there will be deterioration of the vision when the retina will get detached from the choroid itself. Now coming to the one more portion of the middle coat, that is the ciliary body. So this is also very important structure that you will understand when you look at the functions. So this ciliary body, it is nothing but it is the most anterior portion of this, has some 70 to 80 folds and these folds are called the ciliary processes, and there the iris is attached to the middle of the base of the ciliary body. So these ciliary processes, they play a very important role in the secretion of aqueous humor. Posteriorly, the posterior part, it has the ciliary ring in which the ciliary muscle lies, which does the ciliary muscle. This is again one more important structure because the contraction or the relaxation of this muscle is responsible for the one important reflex called the accommodation reflex, which you will be learning in the next class. Okay. So this ciliary body as a whole, it is very important because it has the ciliary process which is involved in the secretion of aqueous humor, and this is also having the ciliary muscle which has a role in the accommodation process. Okay, and this ciliary muscle, it is highly supplied by the parasympathetic fibers. Okay, that means to tell it is under the influence of autonomic nervous system, that is both sympathetic and parasympathetic fibers can regulate the contraction of the ciliary muscle. Coming to the functions of ciliary body, as I told, the ciliary process, the epithelial cells which are present in the ciliary process of the ciliary body is responsible for the formation of aqueous humor. Next, because of the presence of ciliary muscle, it has a role in accommodation response, okay, accommodation reflex. Next, it is also responsible for the secretion of mucopolysaccharide substance and also hyaluronic acid into the vitreous. Vitreous is nothing but one more fluid which is present in the posterior segment of the eyeball. Aqueous humor, it is present in the anterior segment. Vitreous is present in the posterior segment of the eyeball. The outflow, next, the outflow of aqueous humor, it also regulates the outflow of the aqueous humor by regulating the intertrabecular spaces. This you will understand when I discuss about the drainage of aqueous humor. And finally, the cells, the tight junctions which are present in the epithelial cells of the ciliary process, they are involved in the formation of the blood aqueous barrier. Okay, so they are involved in the formation of blood aqueous barrier. So next is this is the diagram showing the different structures of the middle coat of the eyeball. So the most outermost coat is the cornea and the sclera. So here you have the sclera, the whitish portion. Okay, so next this is the conjunctiva, the bulbar conjunctiva, which overlies the whitish layer, that is the sclera. And here you have the middle layer. Observe here. So here comes the ciliary body, which mainly includes two important structures. So ciliary process, it is present anteriorly, and these are nothing but some 70 to 80 folds which are thrown here, and they form the ciliary processes. And this is the ciliary muscle, which plays a very important role in the accommodation reflex. So next comes the iris. Iris is the thin pigmented layer which has a role in preventing the scattering of the light which is entering the eyeball. And next you have the choroid. Okay, so this is the iris, and then it will continue as the choroid on the inner aspect of the eyeball just before the retina. So this is about the middle coat of the eyeball and its function. Now coming to the iris. Iris is the pigmented layer that gives the eye color. You have observed some of the individuals that have a brown colored eye, or some will have a blue color, especially the cats, if you observe, they have multicolored eyes. So it is because of the presence of this pigmented layer, that is the iris. So iris is responsible for the color of an eye of an individual. So this is a contractile disc with a diameter of 12 millimeter. So if you observe this, where is the iris present? This colored portion represents the iris. So this central dark area, what you are seeing, this is just an aperture. Okay, so this is just an aperture, and this is called the pupil, and this white portion of the eye is the sclera. So this is the sclera, and inner aspect of this lower eyelid will form the conjunctiva, and this colored pigmented area is the iris, and this is the pupil. This iris, it has again two sets of muscles, especially the sphincter pupillae and the dilator pupillae. As the name itself says, the sphincter pupillae, it brings about the contraction of the pupil, and dilator pupillae, it brings about the relaxation of the pupil. So they play a major role in the outflow of aqueous humor and also in controlling the amount of light rays entering the eyes. Okay, so these are the important muscles which are present in the iris, and they control the size of the pupil. Okay, and heterochromia, the presence of multicolored iris, can be seen in lower animals like the cats. So coming to the pupil, this you have observed in the previous slide, the diagram. It is a rounded opening present in the iris. Diameter will be about 1 to 8 millimeter depending on the illumination. Because if you throw a ray of light onto the eye, what will happen? The pupil will constrict. Okay, suppose if you remove the light source, what will happen? It will slowly dilate. So the diameter of the pupil varies from 1 to 8. So the maximum diameter is observed when you enter a dark area, and the minimal is observed when you are exposed to a brighter area. So this depends on the illumination, and this regulates the light or allows the central rays, and constriction increases the depth of the focus. So this is mainly to make the light rays just to focus on to the corresponding point of the retina. Okay, and there is normally what will happen in both the eyes, the size of the pupil remains the same, but in some conditions the size of the pupil may be different in the same individual, then it is called anisocoria. So there will be a difference in the pupillary diameter of more than 0.4 millimeter between the two eyes. So coming to the innermost layer of the retina, so this is very important layer because this is the layer which is hosting the receptors for vision, and the receptors for vision are called the photoreceptors. The 72% of the sphere is comprising of the retina, and this is about 22 millimeter in diameter, and it is having 10 layers. Okay, and this is the layer which hosts the receptors called the photoreceptors. Photoreceptors means the receptors which are sensitive to light. They are called the photoreceptors. The photoreceptors are the rods and cones. The posterior part of this retina, it has more cones, whereas anteriorly it has more number of rods. This I will explain in the next slide. Okay, so there is no uniform distribution of photoreceptors on the retina because this is going to determine the proportion of receptors which are present in a particular area is going to determine the visual acuity, ability to identify the details of an object. Okay. Now, apart from these photoreceptors, it also has various the neurons, different types of neurons, especially the bipolar cells, the ganglion cells, the horizontal cells, and the amacrine cells. So these are the different cells which are present in the retina. So this is the diagram of the retina. Probably you would have studied in histology also, and in physiology also this is very important. So this is the external layer and this is the innermost layer of the retina. So from external to internal layer, we have the inner limiting membrane, the first inner limiting membrane, then comes the optic nerve fiber layer, then you have the ganglion cell layer, this is the inner plexiform layer, and this is the inner nuclear layer, this is the outer plexiform, the outer nuclear, and the inner segment. This is the layer of rods and cones, that is the photoreceptors, and finally you have the pigmented epithelium. Okay, finally you have the pigmented epithelium. Suppose when the light ray has to reach these photoreceptors, it has to pass through the limiting membrane first, then the ganglion cell layer, that is then the optic nerve fiber, and then the ganglion cell layer. So it has to penetrate through the ganglion cell layer, then only it can reach the photoreceptors. The electrical activity begins in the ganglion cell layer, and finally it will lead to the genesis of receptor potential. Finally that will be responsible for the development of action potential, which is carried via the optic nerve to the visual cortex. So this is about the structure of the retina, or the different layers of the retina. Normally in humans, actually we can visualize the innermost layer of the retina by using a special instrument called ophthalmoscope. Suppose when you view through the ophthalmoscope, this is the picture we get in the this is the picture we get to see for the retina. So it looks like a reddish globe. Okay, it looks like a reddish orange or the globe, or a bright orange sun, isn't it? So this is the view from the ophthalmoscope, and you visualize the retina through the ophthalmoscope. Now this retina, it has a posterior pole and the anterior pole. The posterior pole of the eyeball, there is a presence of yellowish portion on the retina, which is called the macula lutea. This is the thinnest portion and it has no blood vessels on it. Okay, so the macula lutea, it is present near the posterior pole and it is yellowish in color. Next is the central part of this macula lutea is called the fovea centralis. The central part of the macula lutea is called the fovea centralis, depression with only cones. Okay, fovea centralis is the area where there is presence of only one type of photoreceptors, that is the cones. Okay, so there are two different types of photoreceptors, the rods and the cones. The main function of rods, it is mainly for the dim vision, which is called the photoptic vision, whereas the main function of cones, it is mainly for the daylight vision and also for the increased visual acuity. So if the cones are present, they contribute for maximum visual acuity. That means the person can perceive, or the person can identify the object in a more depth way. That means he can appreciate the details of an object in a more the person can see the object and appreciate the details in a more easy manner. So the cones are mainly concerned with increased visual acuity, and these are mainly present in the fovea centralis. Next, 3 mm medial to the fovea centralis is the presence of optic disc. Okay, so 3 mm medial to the fovea centralis is present. There is a presence of optic disc, which is called the blind spot. Ganglion cell axons converge at the optic disc to form the optic nerve. So it is at the optic disc, the ganglion cells of the retina, they converge and they finally form the optic nerve, which will carry the impulses from the periphery to the primary visual cortex, which is located in the occipital lobe. So what are the other uses of ophthalmoscope? So by using ophthalmoscope, it is a very portable small instrument, handheld device, and usually the examiner will hold the instrument in front of the observers, in front of the subject's eye, and then he can adjust the power of both the eyes, because the observer's means the subject's eye power will be different from the examiner's eye power. So this instrument, it is provided with a series of lenses wherein he can adjust the power of both the eyes, and finally he can visualize the retina. So what all things you can make out by seeing through the ophthalmoscope? to evaluate the retinal blood vessels and to detect the vascular disease. In the previous slide you have observed there are so many vessels, isn't it? All these vessels you can visualize and you can predict or you can diagnose any clinical disorder. Okay, so you will come to know the condition of the eyeball. Next, to detect edema or cupping of the disc, for example, if there is any fluid in the retina itself, so that will lead to the cupping of the optic disc or it will lead to edema. So this is called the pupil edema. The disc, the edema of the optic disc is called the pupil edema. They tell eye is an opener for the brain function. Okay, the brain function can be assessed through the eye. So that is the reason this ophthalmoscope is very important to visualize the retina and to assess the brain functions as well. Next, to identify retinopathy, especially in most common clinical disorders like the diabetes mellitus and hypertension. Okay, so there will be changes in the vessels itself, will be proliferative changes or some changes will be observed in the vessels of the retina. So all these can be visualized by using ophthalmoscope. If the retina is not visual, that means if the details of the retina cannot be appreciated by using ophthalmoscope, then we can go for one more important diagnostic test, that is the electroretinogram. So this is again very important. Usually this records the electrical activities of the retina. Okay, this records the electrical activities of the retina and it will be a biphasic wave pattern usually observed in case of normal individual. Sometimes there may be presence of one more positive wave, that is the C wave also. So this is suggestive of normal electrical activity of the retina. So this is mainly done to diagnose the diseases of the retina and also to detect any congenital retinal dystrophy is there. Even that also can be diagnosed by doing electroretinogram, which is simply called ERG. So that was about the innermost layer. So the structure wise retina is very important. You have to know the 10 layers of the retina in an order. Okay, because when we start discussing about the processing of the vision, you need to analyze how does the light rays pass through the different layers of the retina, and before reaching the layer of photoreceptors, what are the layers that it has to penetrate. Okay, and apart from that, the fundoscopic view, it is very important to diagnose various important clinical disorders. Okay, so that was about the different tunics of the eyeball. Now coming to one more important structure which is present in the eyeball is the lens. So this is the cornea, and here you have the pupil, and pupil is nothing but through the lens there is a presence of some transparent structure which is nothing but the lens, and this lens will be held in its position by a ligamentous structure called the zonules. This is also called the suspensory ligament or the zonules. Okay, so lens, it is a biconvex crystalline structure. It is biconvex. Okay, so this is one convex surface and this is the one other. So this whole thing is the lens itself. So this is a biconvex crystalline structure having the refractive index of 1.42. This is more than that of the cornea itself. Okay, so having a refractive index of 1.42, and this is mainly made up of collagen with collagen fibers along with the elastic capsule. Okay, so this is very important. So in a normal person, if the person is less than 40 years of age group, then what will happen? The lens will be highly elastic in nature. But as the age advances, what will happen? There will be decrease in the elasticity of the lens capsule, which will interfere with the visual process. Okay. Now this lens is held in place by a suspensory ligament which is attached to the ciliary body. Ciliary body, which was the structure present in the middle coat. So this lens will be held or attached to the ciliary body by means of the suspensory ligament, which is also called the ligament of Zinn or the zonules. Now the tautness of the ligaments makes the lens flat. Suppose if these ligaments, if they pull the lens, what will happen? The lens becomes flat. Okay, the lens becomes flat, and loosening of the ligaments will make the lens more curvature. Suppose if they relax too much, what will happen? There will be more bulging of the lens. Okay, so now coming to the applied aspects. As it applied aspects, you must be familiar with the term cataract, because more commonly seen in old age individuals. Normally the lens has to be very transparent and it has to be a crystalline clear. It has to be crystal clear to see the objects very clearly. But as the age advances, because of denaturation process of the proteins which are present in the lens, so there will be this lens, the transparent lens or the the crystalline clear lens will become opaque. So that time the subject's vision will be affected. Okay, and this is called the cataract. Cataract is nothing but the opacification of the lens. So this is more commonly seen in old age individuals. Then it is called the senile cataract. Senile means it is related to the old age. So senile cataract, and sometimes this lens can become opaque because of some trauma or some physical injury, traumatic or physical injury. In both the cases, the lens has to be replaced, and it is done by a surgical procedure called IOL implantation, that is intraocular lens implantation. Okay. Now coming to the fluids which are present in the eyeball, vitreous humor, this is present in the posterior segment. Okay, so this is the posterior segment which is present behind the lens, and in front of the retina is the posterior segment. This entire posterior segment is filled with the vitreous. So this is a gel-like substance behind the lens, and it keeps the eye from collapsing. That means it is a eyeball itself is a cystic structure. It is a fluid-filled sac. But the normal shape of the eyeball, it is maintained because of the presence of these fluids, that is the vitreous and the aqueous humor. Okay, so this keeps the eye from collapsing, and it lasts for lifetime, and it is not replaceable. Okay. Now coming to the chambers of the eye, as I told, there are two different chambers which are present in the eyeball. Anterior chamber, this is mainly present behind the cornea and in front of the iris. Okay, mainly behind the cornea, in front of the iris. Next I will be showing in the diagram. Posterior chamber behind the lens and in front of the retina. So through the pupil, the anterior and the posterior chambers, they communicate with each other. Okay, so normally also there will be a communication between the anterior chamber and the posterior chamber. Now coming to the maintenance of various refractive media of the eye. So you have learned that the cornea itself can act as a refractive media. The lens also acts as a refractive media, the aqueous humor, etc. So the main prerequisite for the visual function is the maintenance of a clear refractive media of the eye. Okay, so the main prerequisite for the processing of vision is the maintenance of the clear refractive media of the eye. Okay, so the main factor which is responsible for the transparency of the ocular media is their avascularity. So because of avascularity, it is contributing for the major transparency, and the structures which are forming the refractive media of the eye from anterior to posterior, or the first comes the tear film which was produced by the lacrimal apparatus, then comes the cornea, a transparent avascular structure, aqueous humor which is present in the anterior segment, and the crystalline lens which is present behind the anterior segment, and vitreous humor which is present behind the lens, that is in the posterior segment. So this was the order of the refractive media of the eye from anterior to posterior aspect. So first you have the tear film, then the cornea, followed by aqueous humor, then the lens and the vitreous humor. So all these they exert some kind of refractive index. So coming to the image forming mechanism. So since the class was started a bit late, I'll come to this later. First let me discuss about the aqueous humor. So this is very important. What is this aqueous humor? Aqueous humor, it is nothing but it is a clear watery fluid. Okay, it is a clear watery fluid which mainly fills the anterior chamber, and the quantity which is present in the anterior chamber, it is about 0.25 ml. Okay, and to some extent it is also present in the posterior chamber, that is about 0.06 ml of the eyeball. So aqueous humor is nothing but a clear watery fluid which is mainly present in the anterior chamber of the eyeball. So look at the diagram here. So this is the cornea. So behind the cornea and in front of the lens is the presence of anterior chamber. So this whole thing is the anterior chamber, be in front of the lens and behind the cornea. So this anterior chamber contains the aqueous humor, the fluid which is present in the anterior chamber. Next, behind the lens and the whole in front of the retina, there is a presence of vitreous humor. Okay, and here you concentrate on this structure, the ciliary body, because here there is presence of ciliary process which is mainly contributing for the production of aqueous humor. Okay, so it will be produced here, then it will be passing through the pupil to enter the anterior chamber, and here it will get spread all over the anterior chamber and it gets filled, and finally it will reach the venous system via the trabecular meshwork which will be present here. Okay, so this is a watery fluid which is present in the anterior chamber of the eye. What is the composition of this aqueous humor? So this aqueous humor, it has 99.9% of water and 99.9% of water, and it also has some of the proteins which accounts for 0.04%, and various ions like sodium, potassium, chloride, they are also present in the aqueous humor. The values in bracket, they represent the millimoles. Okay, 144 millimoles per kg and 4.5 millimoles, 110 millimoles, like that. So this also has glucose, lactic acid, amino acids, and also inosita. So this is the composition of aqueous humor. Now coming to the mechanism of formation of aqueous humor. The mechanism of formation, it mainly involves three important processes. The first one is the ultrafiltration, then comes the secretion, and finally the diffusion process. So these are by these mechanisms the aqueous humor will be formed from the plasma itself, just like your glomerular filtration which was formed by the plasma. In the same way, the aqueous humor will be derived from the plasma. So this is derived from the plasma within the capillary network of the ciliary processes at the rate of 2 to 2.3 ml per minute. So the rate of secretion of aqueous humor, it is about 2.3 ml per minute. Next, what are the different processes involved? It is mainly by ultrafiltration. That means to tell, the plasma substances, they pass out of the capillary wall and the loose connective tissue and also the pigment epithelium of the ciliary process. So just remember, it is the epithelial cells which are present in the ciliary process of the ciliary body. They play a major role in forming the aqueous humor, and the first step is the ultrafiltration. That is it is a filtrate of the plasma. It is derived from the plasma itself. Next, to this filtrate, some of the substances they get added, okay, by the action of sodium potassium pump and because of carbonic anhydrase enzyme. Again, this is one more area. The carbonic anhydrase enzyme is abundantly present in the ciliary process of ciliary body. Okay, because of the action of these two, the pump and the enzyme, some of the substances they get secreted into the filtrate like sodium, chloride, potassium, ascorbic acid, amino acids, and bicarbonates. They all will get secreted into the filtrate. And finally, there will be diffusion. The sodium is primarily responsible for the movement, as you have learned in renal tubular reabsorption. Whenever the sodium is entering, it will drag all the other molecules like bicarbonates, chloride, phosphates, glucose, etc. and also to some extent the water molecules, and this will enter into the posterior chamber where the ciliary body is located. So this is the process of formation of aqueous humor, and this formation of aqueous humor, it is highly under the influence of, or it is highly regulated by the autonomic nervous system, especially the parasympathetic and the sympathetic innervation. The parasympathetic, whenever there is increased parasympathetic stimulation, so there will be increased aqueous secretion. But when there is increased sympathetic stimulation, for example, when the person is in a stressful state, so what will happen? There will be reduction in the production, means there will be decrease in the formation of aqueous humor. Formation of aqueous humor. So the parasympathetic in a way, the parasympathetic stimulation brings about increase in the aqueous humor. So coming to the drainage of aqueous humor. So as you have learned, so this will be formed in the ciliary body. So this is ciliary body, and this is the lens, and here you have the ciliary process, and here there will be formation of aqueous humor. So once it is formed, it will reach the anterior chamber via the pupil. So this is the pupil which is supported by the pigmented epithelium, that is the iris and the suspensory ligaments. Okay, so through the pupil it will enter the anterior chamber and it will get spread over the anterior chamber, and finally it will be drained by the trabecular meshwork which is present present here, okay, and this is the area where the canal of Schlemm is present, okay. So through the canal of Schlemm it will reach the venous, that is the episcleral vein and other venous channels, and finally it will be drained. So the canal of Schlemm plays a very important role for the drainage of aqueous humor, and this is present at the sclerocorneal junction, that is the limbus. So aqueous humor, if you take some books, they show it as the conventional flow and the non-conventional or the unconventional flow. The conventional flow accounts for more than 90% of the outflow of aqueous humor. So what is this conventional flow? So from the ciliary process, the aqueous humor will be formed in the posterior chamber. Through the pupil it will enter the anterior chamber. From the anterior chamber it will go to the trabecular meshwork, and through the canal of Schlemm it will go to the collector channels, which are nothing but the venous channels which are present there, which will open into episcleral veins, and that is how the aqueous fluid will be drained. In the unconventional pathway, this mainly accounts for lesser percentage of flow, that is 10%. And here what will happen, from the anterior chamber, that it will go to the ciliary body, and from there to the suprachoroidal space, and it will reach the venous circulation of mainly the middle layer of the retina, middle layer of the eyeball, that is ciliary body, choroid and the sclera. Therefore this pathway is called uveoscleral outflow pathway, uveoscleral, but this contribution is very less, it accounts for only 10% of the flow. So what are the characteristic features of this aqueous humor? The aqueous humor, it mainly has a specific gravity of about 1.002 to 1.004, and it has a refractive index of again 1.34. So this is again acting as a refractive media, isn't it, along with the cornea, the lens and the other structures. Coming to the functions of aqueous humor plays a very important role in maintaining the normal shape of the eyeball. This I have already explained. Both aqueous humor and the vitreous humor, they contribute for the maintaining the normal shape of the eyeball, because eyeball itself is a fluid filled structure or the cystic structure. Next, it is also responsible for maintaining the intraocular pressure. Now you have seen eyeball is made up of outer covering, inside which is filled with only fluid. So whenever the fluid is there, it will exert, there will be continuous movement of the molecules and they exert the pressure, and this pressure which is exerted here by the fluid is the intraocular pressure, and normally this much pressure is required to maintain the normal shape of the eyeball. So the normal intraocular pressure is about 12 to 20 millimeters of mercury. On an average it is about 15 millimeters of mercury. I repeat, the normal intraocular pressure is about 12 to 20 millimeters of mercury. On an average it is 15 millimeters of mercury. And this aqueous humor, it also acts as a refractive medium and thereby contributing for the process of image formation. Next, it supplies nutrition and drains the metabolites from surrounding structure, because you have learned cornea is an avascular structure. It mainly depends on aqueous humor for its nutrition. So these are some of the important functions of aqueous humor, but I will be discussing more about the IOP, the intraocular pressure. So coming to the role of aqueous humor in maintaining the normal intraocular pressure. IOP refers to the pressure exerted by the intraocular fluids on the coats of the eyeball, okay, because the fluid is present in a closed chamber, that is they are present in the tunics of the eyeball, and they continuously exert some amount of pressure on the coats of the eyeball, and this is called the intraocular pressure. The normal IOP varies between 10 to 21 millimeters of mercury or 12 to 20 millimeters of mercury. Various books gives the ideal range is 12 to 20 millimeters of mercury. On an average it is about 15 millimeters of mercury, and this is mainly maintained by the dynamic equilibrium between the formation and the outflow of the aqueous humor. So when the formation is normal and the outflow is occurring normally, then only the IOP will be maintained at 10 to 21. Suppose if there is increased formation than the outflow, or suppose there is decreased outflow than the formation, both conditions can alter the intraocular pressure. So what will happen if there is persistent increase in the intraocular pressure? So this will lead to a clinical condition called the glaucoma. So most of you have heard about this clinical condition. All these conditions will be learning in detail when you go to final phase, that is in ophthalmology. But now as a part of applied aspects, you must know what is this glaucoma. So clinically, actually we can measure the intraocular pressure by using a special instrument called the tonometer. This is again a small handheld portable instrument. So this will have a flat base like your tuning fork, the base of the tuning fork. So that flat end of the tonometer is placed over the anesthetized cornea. They will place some anesthetics and it will be placed over the cornea. Then they apply pressure, okay, they will apply the pressure over the cornea. So what will happen? So there will be indentation, there will be inward movement of the cornea, and by assessing this they can predict the intraocular pressure. So that is how they measure the intraocular pressure by using an instrument called tonometer. The most widely used tonometer is Goldman's tonometry. Okay, so that is commonly used to measure the pressure within the eyeball. What is this glaucoma? So glaucoma, it is a degenerative disorder which is mainly characterized by loss of retinal ganglion cells. The main cause for this glaucoma is the raised intraocular pressure. So here the pressure, if it increases more than 25 to 30 millimeters of mercury, it will cause permanent damage to the retinal layer. That means there will be loss of retinal ganglion cells. In the beginning itself I told, the light rays first they have to reach the ganglion cells to initiate the electrical activity in the retina. So if there is damage to the ganglion cells, there will not be any processing of the vision. Okay, so if the pressure is more than 25 to 30 millimeters of mercury, then we call this condition as glaucoma. And glaucoma can, as it is causing permanent damage, structural damage in the retina itself, it will lead to blindness also. The person will not be able to see. And if the pressure, if it increases suddenly to more than 30 millimeters of mercury, it can lead to death of an individual also. So what are the different types of glaucoma? Clinically we have two different types of glaucoma based on the cause. So they are called open angle glaucoma and the angle closure glaucoma. Here the angle is referred to the canal of Schlemm, which is present at the sclerocorneal junction, that is the limbus. So open angle glaucoma, this is mainly because decreased permeability to the trabeculae itself. Here the canal of Schlemm will remains open, but what will happen? There is decreased permeability through the trabeculae. So finally what will happen? Aqueous humor, once it is formed, it will be entering the anterior chamber and finally it will be drained by the trabecula which is present at the limbus, isn't it? So these trabeculae, they will not be able to reabsorb, they will not be able to take out the fluid, take out the aqueous humor, isn't it? So that is contributing for increased pressure within the eyeball, and this is called open angle glaucoma because the angle of the canal of Schlemm remains open. Next is the angle closure glaucoma. So this angle closure glaucoma means it is mainly related to the width of the canal of Schlemm. So this is mainly observed whenever there is forward movement of the iris which is closing the angle. So whenever the forward movement of the iris or when there is increased curvature of the means, the constriction of the pupil as well as the dilatation of the pupil can affect the patency of the canal of Schlemm. So that time it can lead to angle closure glaucoma. So these are the two different types of glaucoma depending on etiology. So how can we treat this clinical condition? So as I told, in the secretion of the aqueous humor, the carbonic anhydrous enzyme, it is playing a major role in the secretion of the fluid, and this is the one which is facilitating the ultrafiltration of the plasma also. So if we use carbonic anhydrase inhibitors, okay, so that time what will happen? We can actually decrease the secretion of aqueous humor. Apart from this, we can actually use the cholinergic agonists like physostigmine or physostigmine. This you have heard in case treating the myasthenia gravis. The cholinergic agonists like physostigmine and pilocarpine, they bring about the dilatation of the pupil, or they mainly alter the size of the pupil and they facilitate the flow. And the last remedy, if the medical line of treatment, if it doesn't respond, then we have to go for the surgical line of treatment. So here what they do, they make incisions in the trabecular meshwork, and through the trabeculae the fluid can easily enter into the venous sinuses, and that's how it can get drained. Okay, so this is about the treatment modality for the glaucoma. Now coming to the MCQs. The first question, when the light passes through the lens, the system of the eye, it first encounters. So So which is the answer here? So this is the first inner limiting membrane. Okay, the layer of rods and cones, they are present far ahead, they are like situated more on the inner aspect. Okay, so the first it has to fall on the inner limiting membrane, then it will reach the ganglion cell layer, and finally it will reach the layer of rods and cones where there is genesis of receptor potential. So next, the photoreceptors present in fovea centralis. Fovea centralis, it marks the area of maximum visual acuity. So whenever the visual acuity is more, obviously it is the presence of codes. Okay, codes are responsible for main visual acuity. Then the structure forming refractive media of the eye from anterior to posterior aspect are, so first you have the teophyll, then you have the cornea, then you have the aqueous humor and the lens and the vitreous humor. So the teophyll, the cornea, then the aqueous humor, then the lens and the vitreous humor. So B is the answer. This is the order. Then the next question, the normal intraocular pressure of the eye is about 12 to 20 millimeters of mercury. On an average, it is about 15 millimeters of mercury. The aqueous humor, it is mainly secreted by the epithelial cells of the ciliary process of the ciliary body itself. So please read about the functional anatomy of the eyeball, because if you understand this, it will be easier for you to understand the next classes. Okay, so read about all this, practice drawing the diagram and labeling all the different layers of the eyeball. Okay, so thank you. Any doubts, I'll respond in the group.
morning all of you. So a few of you had asked me doubts regarding transcription termination, where in prokaryotes I said there is row dependent factor and row independent factor. This video will actually simplify your doubts. There is something called row factor dependent, and the other one which I said was it will be in a sequences. There will be some sequence which will terminate forming a hairpin like structure, right? So you can see this in this video.
First, I wish to talk about the termination of transcription in prokaryotes. This can happen in two ways, either row-dependent or row-independent termination. First, I will discuss row-independent, also known as factor-independent termination. This type of termination occurs when the messenger RNA contains an inverted sequence of bases that pair with each other to form a hernia structure. This hernia is normally rich in the cytosine and guanine base pairs, as they are joined together by three hydrogen bonds between the adenine and uracil. This gives the structure of greater resilience. Following this formation, the RNA polymerase associates and transcription termination is almost complete. The other type of termination involves a row protein, which is an ATP-dependent nuclease. This protein binds to the C-rich region of the mRNA strand and moves from 5' to 3' direction towards the transcription pupil. RNA polymerase stalls at the GC-rich region, giving the row protein sufficient time to catch up. Row unwrites the DNA-RNA hybrid, and then row RNA polymerase and mRNA all release from the DNA strand. Less is known about transcription termination in eukaryotes. Transcription termination
Okay. So that was about termination of transcription in prokaryotes, where I said there is one factor, row factor dependent and row factor independent, right? In row factor independent, there are certain sequences which form a hairpin like structure, and then the RNA polymerase and the RNA strand is released. But in row factor, you had seen the video earlier, yesterday I had shown, the factor goes and binds to the DNA template strand, and so the RNA polymerase and the RNA strand will be released. Okay, that is about termination. Next we will move on to post-transcriptional processing. Now what is post-transcriptional processing is whatever transcript, after transcription from a DNA we get a RNA, right? What we get from RNA, that is called as primary transcript. Okay, that primary transcript is not mature enough to, or what you call it as, it is not in an activated form. So to make it an activated form, this post-transcriptional processing should happen. The steps involved are either of these things, or two or more things can happen in one of the things to activate the transcript. Okay, one is endonuclease cleavage. If there is any extra bases or anything is added, there can happen endonuclease cleavage, or poly-A tailing. A is nothing but adenine. Addition of adenosine group at the end is called as poly-A tailing, or there can be 5' capping, where we call it as at the 5' end there can be capping by a guanosine or any other bases. Okay, or there can be methylation, addition of methylation group at the 5' end or hydroxyl group. Okay, and there can be removal of introns. There will be exons and introns. I will be showing that how the removal of introns and splicing of exons happens. So these are all the steps which can happen to for the post-transcriptional processing, where a primary transcript can be primary transcript is not in an activated form. What we get from DNA template, what the RNA we get from DNA template, it is not in the activated form. Okay, to convert it into an activated form, this post-transcriptional processing should happen. After the transcript is obtained, this process starts. Okay, so when an mRNA is formed and released from the DNA template, it is known as primary transcript. That's what I said. Whatever from the DNA directly, the transcript, whatever you are getting, that we call it as primary transcript. Okay, and it is also known as heterogeneous nuclear RNA or hnRNA. Okay, the mRNA formed and released from the DNA template is known as primary transcript, and it can also be called as hnRNA or heterogeneous nuclear RNA. Okay, now this whatever primary transcript is obtained, they undergo modifications to form a mature RNA or the activated form. Now what did I tell? The modifications can be through endonuclease cleavage, poly A tailing, 5' capping, and removal of methylation and removal of introns and splicing of exons, right? So in endonuclease cleavage and poly A tailing, there will be few nucleotides from 3' end are cleaved and many nucleotides like A adenosine nucleotides are added. So what happens exactly in endonuclease cleavage and poly A tailing is some of the nucleotides are removed from the 3' end. Okay, and they are cleaved and and many of the adenosine group A base is A nucleotides are added, and that is called as endonuclease cleavage or poly A tailing. Now when capping that 5' end it is capped by 7-methyl guanosine triphosphate. Okay, methylation is internal nucleotides of mRNA are methylated. How is removal of introns done? Primary transcript consists of coding regions, that is called as exons. Whatever the coding regions are present, that will be exons, and whatever the non-coding regions will be present, because you know in a DNA what is happening, not whole of the DNA is converted into RNA. There are few sites which are selected as promoter site, and that is being forming RNA, right? So whatever the gap, whichever is coding regions, and there will be non-coding regions. There will be exons and there will be introns. These intron sequences should be removed and the exons should be spliced or combined together to form a activated RNA. So you can see here the heterogeneous RNA or the primary transcript, they undergo end modifications where there can be a capping, that is 7-methyl adenosine guanosine triphosphate is added as at the 5' end, that is called as capping, and on the other end there can be poly A tail, that is adenosine nucleotide triphosphates are added. And in case of splicing, there can be in between the whatever introns are there, which is non-coding regions are there, those are removed, and whatever cut pieces, that is called as cutting, and the chemical modifications is new chemical groups are added to activate it. Okay. You can see here, this is how it will be. The primary transcript will be like this. There will be some of the coding regions, there will be some of non-coding regions, and these non-coding regions are spliced out, and whatever exons are there, they are joined together to form a matured mRNA. So whatever the non-coding DNA or RNA of unknown function, that is the introns are removed and the exons are joined together. Okay. So that is one of the post-transcriptional modification of mRNA. Now we learned about how a mature mRNA is messenger RNA is produced. That's how, then the same way we will see for rRNA and tRNA also. There will be post-transcriptional modifications happening for rRNA and tRNA also. There is something called small nuclear RNAs, and these are about and they are also called as NUCs. There are about 1 million copies of small nuclear RNA per cell. Okay, the small nuclear RNAs, they are numerous in amount. They are almost 1 million copies of small nuclear RNAs are there in a cell, and they are named as U1, U2, U4, U5, U6 and U7, where it is uracil rich. Okay, the U stands for uracil rich nature of small nuclear RNA. So what is The small nuclear RNA is made up of, they are made up of many uracil group. Okay, so it is rich in uracil group. Okay, so how many copies of small nuclear RNAs you find in a cell? It is about 1 million copies of small nuclear RNAs are found in a per cell. What is its main function? Why should a small nuclear RNA be present, or what does it help in? They take part in the formation of spliceosomes. Now you learned what is splicing. It was just the introns were removed and the splicing of exons were done, or the joining of exons were done. These spliceosomes, they take part in the formation of spliceosomes. Okay, the small nuclear RNA, they bind to a specific protein to form a nuclear ribonucleoprotein particles. Those are called as SNPs. Okay. Now what is this small nuclear RNA doing? They actually bind to a specific protein and they form small nuclear ribonucleoprotein particles, which are called as, which are even, it can be pronounced as SNPs. Okay. So if there is what does this SNPs do? They associate with the heterogeneous RNA at the exon-intron junction and they form a spliceosomes. Okay, if there are any production of antibodies to this against this SNPs, against the SNPs, they cause systemic lupus erythematosus, which is a autoimmune disorder, where there is immunity to the autoimmune is immunity, it is showing immunity to the self cell. It is defending its own cell. Okay. So this is what happens. Now what is this small nuclear RNA doing? There are many million copies of small nuclear RNAs in the cell and they are named as U1, U2, U4, U5, U6 and U7. And what does U stand for? It stands for uracil rich in nature. What the main function of small nuclear RNA is, they take part in the formation of spliceosomes. How do they take part in the formation of spliceosomes? They actually bind to the specific proteins to form a small nuclear ribonucleoprotein particles, which are called as SNPs. Okay. Now if there are any production of antibodies to this SNPs, like what is SNPs should actually remove the introns, right? If there is any antibodies produced, it causes a disease called systemic lupus erythematosus, and these SNPs are associated with the heterogeneous RNA, that is the primary transcript at the exon-intron junction. I will show it in the picture. Now you can see it here. This is the DNA, where there will be some of the coding region, non-coding regions, coding regions, non-coding regions and all that. From this DNA, RNA is being produced, that is called as transcription, which will again have exons, introns, that is the coding and non-coding regions. Now this non-coding region is removed and the exons are joined together to form from a primary transcript, it forms a messenger RNA, that is a mature RNA. Okay. Now this pre-mRNA, here you can see, these are the SNPs. These SNPs, what do? They come in between the junction of exons and introns and there will be a loop-like structure formation, and next it removes the whatever excised intron in a lariat shape, it is removed and the spliceosomes are, and the SNPs are released and there is formation of mature mRNA. So SNPs and spliceosome can be a question for you. You will have to write about this, how it actually removes the introns from the exon-intron junction and how a mature mRNA is formed. Okay. So that is one part of post-translational modification. I repeat. So under post-translational modifications, there can be processing, there can be endonuclease cleavage, poly-A tailing, 5' capping, methylation, removal of introns and splicing of exons. So if we take messenger RNA for the first, whatever messenger RNA is formed and released from the DNA, that first part is called as primary transcript, and it is also called as hnRNA or heterogeneous nuclear RNA, and they undergo several modifications to become mature RNA or to form a active form, right? Now what is this modifications in this messenger RNA for the pre-mRNA to be converted into mature mRNA? What is happening? First thing is endonuclease cleavage and poly-A tailing, where the few nucleotides from the 3' end is cleaved and many A nucleotides are added, and there can be capping at the 5' end by 7-methyl guanosine triphosphate. Methylation, that is internal nucleotides of mRNA are methylated, and there will be removal of introns. Primary transcript consists of coding regions which are called as exons and non-coding regions or the function unknown regions which are called as introns. Okay, now these intron sequences should be removed so that the exons are combined together or spliced together. Okay, so in mRNA what is happening? It is just excision of introns. How is the excision of introns done? By the small nuclear RNAs. How many small nuclear RNAs are present? There are about 1 million copies, and they are named in numbers are done in U because U stands for uracil, and your small nuclear RNAs are uracil rich in nature. Okay. How do they take part in spliceosomes? They actually bind to a specific protein to form a small nuclear ribonucleoprotein particles which are pronounced as NURBs, and these NURBs, how do they go? They actually bind at the exon-intron junction and they form a spliceosomes. If there is any production of antibodies against the NURBs, it causes a disease called systemic lupus erythematosus. Just to revise this image, you can see there are several exons and introns where the introns are spliced. How is it spliced? This SNRNPs, that is the SNRPs, they bind in the junction of exon and intron, and they remove the intron in a lariat shape, and the small nuclear RNAs are removed, and there will be a formation of mature RNA.
Eukaryotic cells must process the RNA product of transcription to form a mature translabel messenger RNA. The first step in RNA processing is capping, the addition of a special nucleotide to the 5' end. The second step is polyadenylation, the addition of a long tail of A's at the 3' end. A eukaryotic gene typically consists of both exons, regions that encode parts of the protein product, and introns, regions that are not represented in the product. The introns are transcribed along with the exons, but they are spliced out as the third step in eukaryotic RNA processing. Ribonucleoprotein assembly is called spliceosomes due to splicing. The result of 5' capping, 3' polyadenylation, and RNA splicing is an mRNA ready to be translated.
Just a short video to just revise what we learned till now. Next, we will see what are the differences between the prokaryotic and eukaryotic transcription. Okay, in the slide I am explaining the differences between prokaryotic and eukaryotic transcription. Where is it happening? Both prokaryotes and eukaryotes, the transcription is happening in nucleus. Okay, and what is the enzyme? DNA dependent RNA polymerase in prokaryotes, and in eukaryotes there are several other factors also, right, like RNA polymerase 2 for mRNA transcription, RNA polymerase 1 for rRNA, and RNA polymerase 3 for tRNA. I repeat, here in prokaryotes DNA dependent RNA polymerase, that's all. But in eukaryotes, it is RNA polymerase 2 for mRNA transcription, RNA polymerase 1 for rRNA, and RNA polymerase 3 for tRNA. Now, how is the initiation happening in prokaryotes? The sigma factor in the RNA polymerase, it binds to the promoter site. But what happens in eukaryotes? There will be, it requires several transcription factors, and there will be Tata binding protein. I will be asking questions at the end of the class. This differences between prokaryotic and eukaryotic transcription. So please concentrate. What are the start signals for prokaryotes? It is Pribnow box, and in eukaryotes it is Hogness box. How is the termination happening? Termination may be row factor dependent or row factor independent in prokaryotes, and termination may be row factor dependent or independent, but we are not clear how this termination is happening in RNA of eukaryotes. Okay. In the post-transcriptional modification, how is it in prokaryotes? mRNA is transcribed immediately and translated without any modification. Remember this. In prokaryotes, does not require any post-translational processing. They can just directly be converted, translated. But in eukaryotes, it undergoes several post-transcriptional modifications like 5-methyl capping, poly-A tailing, splicing in the nucleoplasm. Okay. Now, what are the actions of antibiotics? You remember this, but I will be teaching it again. It is inhibited by actinomycin and rifampicin, but in eukaryotes, it is inhibited by actinomycin and alpha amanitin. This is again a revision. This DC box, cat box and all are promoter site, Tata box, promoter site. Start of transcription, there will be exon, intron, and there will be post-translational modification. This is just one picture to summarize everything we learned. This is again the same. So we will always in the first picture you can see there will be formation of pre-mRNA. How is it formed? There will be a template DNA strand which will contain exon intron junction and all that, and there will be the region between the promoter and termination sequence, we call it as transcript region, right? And after once the transcription is over, there will be post-translational modification by these steps like 5' capping, and next is poly-A tailing, and there will be splicing of introns, and the exons are together formed, and finally we get this with this. This is the mature mRNA which is going from nucleus to the cytoplasm, where there is just start codon and poly-A tail and 5' end. Don't get confused with this. It is just summarizing the whole transcription and post-transcriptional modification. Now, what are all the inhibitors of RNA synthesis or inhibitors of transcription? It is this antibiotics like actinomycin D and mitomycin. How do they inhibit transcription? They intercalate with the DNA strands. That is, these actinomycin D and mitomycin, they go and bind in between the DNA strands, so that the proper transcription is not at all happening and the transcription is blocked. So they are used as anti-cancer drugs. Actinomycin D and mitomycin are used as anti-cancer drugs, where they block the transcription completely. Okay, and we have rifampicin, which is used as antitubercular drug. It binds the beta subunit. What was the function of beta subunit of RNA polymerase? It was to continue the transcription, right? So it goes and binds there, and hence there will be no transcription happening. So this is widely used in the treatment of tuberculosis and leprosy. Okay, and we also learned about another alpha amanitin. It inactivates the RNA polymerase 2. So there will be no synthesis of rRNA. There will be no synthesis of ribosomal RNA. So what is alpha amanitin? It is a poison which is obtained from mushroom. That's why we usually carefully eat mushroom, right? We need to know which type of mushroom to be eaten because of this thing. It inactivates the RNA polymerase 2, and there will be no production of rRNA, of the ribosomal RNA. Now, what are ribozymes? Ribozymes are the biocatalysts which are made up of RNA, so it is called as ribozymes. It is a biocatalyst. Now, what do they actually do? These are catalytic RNA molecule which sequence the specific cleavage activity, and they have high catalytic, high fidelity, high specificity and low toxicity. These ribozymes are, what are ribozymes? Ribozymes are the biocatalysts which are made up of RNA, and so it is called as ribozymes. What what does it do? It actually these are the catalytic RNA molecule with sequence of specific cleavage activity. Okay, they have high catalytic activity. It has also high fidelity and specificity, but they have low toxicity. Now, this spliceosomes, what we learned, they contain ribozymes, or the small nuclear RNA. They contain the ribozymes. What is its clinical importance is, the hammerhead ribozymes are RNAs that hybridize to complementary RNA sequences. That is, for whatever such ribosomes, whatever is formed, they are specific to the HIV genes and are efficient in reducing the viral protein expression. Now, what they do is, the ribozymes, with the hammerhead of this ribozymes, they hybridize to the complementary RNA sequence. They form hybrid with the complementary RNA sequence, and what does this do is, in specific to HIV genes, they reduce the viral protein expression. Since ribozymes can recognize and cleave the target RNA, they are tried in gene therapy to decrease the translation of proteins. Okay. So this is one 3 marker or 5 marker you can have. Ribozymes, they are the biocatalysts which is having high catalytic activity, high fidelity, high specificity and low toxicity. It actually acts as a spliceosomes, which the spliceosomes contain ribozymes, but here the main use it is doing is it hybridizes with the complementary RNA sequences, and so these ribosomes are very specific to the HIV genes and they are efficient in reducing the viral protein expression. Ribozymes are used in reducing the viral protein expression, and they can recognize and cleave the target RNA, so they are used in gene therapy, mainly to decrease the translation of proteins. That is, from transcription to translation, it is blocking there. There is something called reverse transcription. So, what is transcription actually? There will be synthesis of RNA from a DNA. Now, if I call reverse transcription, what do you think it is? As the word itself tells, there will be reversal of transcription. So, what is it? What is it? Can I see any comments? What is reverse transcription? Any guesses? Transcription is synthesis of RNA from DNA. Right? So, what is reverse transcription? What can it be? At least one of you just make a wild guess. What is reverse transcription? If you know the transcription, it is just reversal of transcription. What can it be? It is DNA from RNA, right? Right guess? So, this is the process of RNA directed synthesis of DNA. Okay, is it happening in human? Where is DNA synthesized from RNA? Is it happening in human? But in human I said it is DNA from DNA, that is replication. Where is this synthesis of DNA from RNA is happening? In which organism? Retrovirus, very good. So, we shall go into the class. So, this is the process of RNA directed synthesis of DNA, which is catalyzed by the enzyme RNA dependent DNA polymerase or reverse transcriptase. So, what is it? It is a synthesis of DNA from RNA by the enzyme RNA dependent DNA polymerase or reverse transcriptase. What is happening? Usually the genes are seen in DNA and DNA dependent RNA polymerase copy the information of DNA to mRNA. But the genetic materials of some animal and plant viruses are made up of RNA. Usually what was happening, the DNA used to contain all the genetic information and then it was transcribed into RNA to form a protein, right? So, but in some animals and viruses, we see animal and plant and viruses, we see the genetic materials are in RNA. That is, example is retrovirus, as few of you mentioned. It is human immunodeficiency or virus or AIDS. It is a retrovirus where there is reverse transcription, where the RNA contains the genetic material and it acts as a starting material to synthesize the DNA. So here the RNA acts as a template. Based on the base sequence of this RNA, reverse transcriptase enzyme will make a new DNA strand. Okay. Now, is the RNA single stranded or double stranded? Is the RNA single stranded or double stranded? Answer, is RNA single stranded or double stranded? Yes, it is single stranded. So, if RNA acts as a starting material, we get a DNA of double stranded. How? How do we get a double stranded DNA? Reverse transcriptase makes a new DNA strand, that is, one strand of DNA is formed. Then there will be from this RNA DNA hybrid, the RNA part is hydrolyzed by specific RNAs, that is the whatever hybrid is formed, that is being spliced up and it is being removed, and then the remaining DNA, one strand of DNA, that acts as a template to produce double stranded DNA, and this process is catalyzed by DNA polymerase 3. I repeat, now the RNA is single stranded. Whatever new strand is complementary, which is forming the new DNA strand, that will be single strand which is being formed, and whatever this RNA DNA hybrid is there, that is being hydrolyzed by specific RNase H, and once this is being removed, the remaining DNA strand that acts as a template to produce another strand, that is the double stranded DNA is formed, and this process is catalyzed by DNA polymerase 3. Thus, the genetic information here is transferred from RNA to DNA. You can see here, this is a 3' to 5' virus RNA, where it undergoes polymerization by reverse transcriptase. Okay, so this is the RNA DNA hybrid. The RNase H, it hydrolyzes the RNA and the DNA copy of RNA is here. Now this DNA copy of RNA, it acts as the template for the other strand to be formed, and this is being catalyzed by DNA polymerase. So we are seeing almost 3 enzymes functioning here. One is the reverse transcriptase, which is synthesizing one DNA strand, and the DNA RNase hybrid is being hydrolyzed by RNase H, and copy of the DNA copy of RNA, it forms a double stranded DNA by the enzyme DNA polymerase 3. What are the 3 enzymes involved here in synthesis of DNA from RNA or the reverse transcription? Which are all the enzymes? Which are the 3 enzymes which is involved? That is a question. Which are the 3 enzymes? It is already there in the image. What are the 3 enzymes involved in reverse transcription? RNase H hydrolases, yes, then DNA polymerase 3, then which is the main first enzyme, reverse transcriptase, right? Good. So also some of the tumor viruses were also shown to have reverse transcriptase. Whenever there is presence of enzyme reverse transcriptase, it may indicate the presence of retroviral infection. Okay. So that was about mRNA, which was produced, the primary transcript of mRNA, which was converted into mature RNA, and we learnt about SNPs, we learnt about spliceosomes, we learnt about ribozymes, and then we also learnt about reverse transcription. Next we will move on to how rRNA and tRNA is synthesized. Okay, that is the ribosomal RNA and transfer RNA synthesis is the next thing. Okay, so if we see a pre-rRNA transcript, there we saw in mRNA, pre-mRNA transcript contained introns, exons and all that, but here it looks like this. This is the 16S subunit, this is the tRNA, this is 23S, this is 5S, and these are the non-coding regions which should be removed. Okay, so first thing is here it undergoes methylation, that is the methyl groups are added in between the nucleotides. Okay, then there will be cleavage of RNases, that is whatever the non-coding regions are removed by the nucleases, then it will be produced in subunits. Okay, this is the 16S ribosomal RNA and the tRNA is produced, and 23S ribosomal RNA and 5S ribosomal RNA. The ribosomal RNA, for the synthesis of ribosomal RNA, there it was in mRNA, it was removal of introns by SNPs, right? So here and spliceosomes, it was joining the exons, but here it is in synthesis of ribosomal RNA, it is the methylation which happens as a first step, and then there will be cleavage of non-coding regions, and then there will be formation of ribosomal RNAs in subunits like 16S ribosomal RNA, tRNA, 23S ribosomal RNA and 5S ribosomal RNA, that is the mature RNAs. Now here we saw there will be production of tRNA, right? How does this undergo post-translational processing? I'll show you. So in a structure of tRNA we have 4 arms, that is the acceptor arm, actually 5 arms, that is the acceptor arm, anticodon arm, DHU arm of tRNA and pseudouridine arm of tRNA. Also there is one thing called as short arm. Okay. So now here you can see this is the tRNA primary transcript which we have got, where there is in the acceptor arm, this will be the acceptor arm at the 3' end, and this will be the anticodon arm, and this will be the DHU arm, and this will be the pseudouridine arm, and this is the short arm. Okay, so you can see here, this is the primary transcript which is containing many other bases and other base sequences are all there. For the post-transcriptional modification to happen in a tRNA, there should be base modification which should happen, and also there should be 5' cleavage and 3' cleavage, and there should be addition of CCA. Okay, what and all should happen for a tRNA from primary transcript to form a mature tRNA? It should undergo a few base modifications, like you can see here from GUU and all that. There is modification to D, that is the DHU arm, which contains dihydroxyuracil. Okay, and there should be 5' cleavage, where there is extra bases here. There should be cleavage of 5', and there should be 3' cleavage here, and there should be CCA addition. Okay, this is the intermediate form before the mature form is formed. This is the intermediate form where there is a acceptor arm, which I said, this is the acceptor arm which is accepting the CCA. Okay, cytosine, adenine. Okay, and there is cleavage at the 5 prime cleavage, that is 5 prime cleavage. Here 3 prime cleavage has happened and there will be addition of CCA, and here there will be modification of bases, that is from uracil it is modified into dihydroxyuracil. Okay, and this is the short arm. Okay, and this is the pseudo-uridine arm. Okay, this is the pseudo-uridine arm. So you can see even T here. This is all, instead of UU there is TU, pseudo-uridine. Okay, and here there should be cleavage of this one also to form a anticodon loop. Finally, the final mature RNA looks like this. This is the acceptor arm. This is 5 prime cleavage happened, and this is the DHU arm, short arm, anticodon arm, and the pseudo-uridine arm. Structure of tRNA is a question for you guys. Okay. So just to repeat about rRNA and tRNA. In ribosomal RNA, there should be, what is the post-translational modification happening to the pre-rRNA transcript? There will be methylation. Methylation is addition of methyl groups at the in between the bases, and there will be cleavage of non-coding arms to release several subunits of rRNA. It undergoes several modifications. Ribosomal RNA usually undergoes several modifications to form a mature RNA. In tRNA, the structure of tRNA will be a question for you, and what is the post-translational modification should happen? There should be a base modification, there should be 5 prime cleavage, there should be 3 prime cleavage, and there should be CCA addition at the acceptor arm. So which is the acceptor arm? This is the acceptor arm, this is the anticodon arm, and here is the DHU arm, short arm, and pseudo-uridine arm. So when we ask for a structure, you will have to write this structure. Okay, structure of tRNA, and if the tRNA processing by RNAs or splicing is asked, you will have to write the whole thing, primary transcript, how the intermediate is formed, and how mature tRNA is formed. That is all about post-transcriptional modification or post-transcriptional processing. So what and all we saw? We saw about 5 prime capping, we saw about endonuclease cleavage, poly-A tailing, methylation, and removal of introns, splicing of exons, right? And we also learnt about ribozymes, reverse transcription, the clinical importance of ribozymes, how is the RNA synthesis is inhibited, that we learned, and the differences in prokaryotic and eukaryotic transcription and post-translational modification, post-transcriptional modification. So if you have any doubts, you can post your doubts now regarding, it can be regarding transcription, replication, post-transcriptional modification. Any doubts regarding that, please post your doubts now. Any doubts regarding replication or transcription or post-transcriptional modifications? If you have any doubts, post your doubts now. Okay, can somebody answer what is SNPs? What is small nuclear RNAs? What are the functions of it? The functions of spliceosomes, it is to join the exons. Can anybody post the functions of SNRPs? And what is it rich in? Small nuclear RNA which helps in removal of introns, right? It takes part in formation of spliceosome. It is rich in uracil, right? Good. So, what are the steps that can involve in post-transcriptional modification? What and all modifications can happen for a primary transcript to convert into activated form? It's already in the image. What and all, post-transcriptional processing can happen for a primary transcript to convert it into a activated form? Splicing of what? Capping, yes. Where? Capping, tailing, splicing. Okay, now expanded where the capping is happening, where the tailing is happening, and what tailing is happening, and what splicing. Endonuclease cleavage, right. Polyadenylated tail, right. What capping? Well, what is that? Where is the capping happening and which one is capped? What is that? Which nucleotide? No attendance given now. I am asking questions. Yes. Poly A tailing at the 3' end, 5' end the capping is happening, yes. Endonuclease cleavage, right. Removal of introns and removal of exons. Fine. So, 7-methylated guanosyl triphosphate, right. Good. So, what are the differences between eukaryotic and prokaryotic transcription? What is the difference between eukaryotes and prokaryotic transcription in the initiation? Ribnoboxin prokaryotes, Hogness-boxin eukaryotes, right? Good. Yes, it is different. How is it different? Enzymes used are different. How is it different? Post-translational transcriptional modification occurs in eukaryotes and it's not known. No transcriptional modification in prokaryotes, right? Enzymes, tell about the enzymes. How is it different in prokaryotes and how is it different in eukaryotes? RNA polymerase 1, 2 and 3 in eukaryotes, right? And in prokaryotes it is DNA dependent RNA polymerase. Very good. So what are the 3 enzymes in reverse transcription? What are the 3 enzymes which is involved in reverse transcription? reverse transcriptase, RNase H, DNA polymerase 3. Very good. So I hope all of you understood the whole class for today, and thank you. If you have any doubts, post in the group. We will move on to translation in next class.
friends. So before we start off with this topic related to the inguinal canal, so you have to hold back patients for about another 1 hour, so that by that time, after 1 hour at least, you can have your lunch leisurely. So till that time, so I request you to hang on with some patience for say about 1 hour. So as I told you, now one of the topic in relation to the abdomen, so the anterior abdominal wall. So rather, so that is, this is the inguinal canal. Now we will be learning the inguinal canal under these following headings. As so now from examination point of view again, this can be a 5 marker or can be asked as a 10 marker as well. And one of the part of this particular topic, that is the inguinal canal or the Hasselbalch's triangle, this itself can be a separately a 5 marker question. And the applied aspect related to this particular topic, that is the inguinal hernias, so that those can be asked as a 3 marker question. So quite an important topic. So we'll see to all of those things in a slightly systematic manner. So first we'll begin with the introductory part. So what exactly is the inguinal canal? So as it is evident from the diagram, so you can make out where exactly the inguinal canal has been situated over here, as you can make out from the diagram here. Now this is considered to be, now if you wish to write an introductory statement to the inguinal canal, so it has to be written as an intermuscular space. So an intermuscular space, or the space present between the muscular layers of the anterior abdominal wall, which you are already familiar, related to the muscles which are present in the anterior abdominal wall. So in between those muscles there is a space. So it is roughly 4 centimeter long, approximately 4 centimeter long, as you can make it over here. So this is the actual situation. Now situated more precisely above the middle. Now if you see from this diagram again, this represents the anterior superior iliac spine, and this represents the pubic tubercle, and the connection, or rather the attachment that has been shown in a darker line, this represents the inguinal ligament. So this canal, that is inguinal canal, lies just about or immediately above or runs almost parallel to the medial part of that inguinal ligament over here, medial half. So that is how, just about the medial half of the inguinal ligament. So that is how you can write an introductory statement to the inguinal canal. Whereas after that, now this is again image just to make you understand in a better possible way. So the inguinal canal lies over here in this particular region. You can make out anterior superior iliac spine, though pubic tubercle is not that clearly seen in this diagram. So this is how the location where it will be located. Now these are the anterior abdominal muscles, which you are, I hope that you are already very much aware of these muscles. That is related to, if you go from outside inwards, the external oblique muscle, the internal, and the transverse abdominis muscle, so which are seen on either side of the abdominal. Apart from that, you can even make out one longitudinal muscle, which is the rectus abdominis muscle. So these are the muscles which are with respect to the anterior abdominal wall. So the inguinal canal lies as an oblique passage in this particular region. Now we'll further look into the exact extent of extent of this inguinal canal. So it is said to be, it is said to be extending between the 2 openings. Now this is called as the deep inguinal ring, and this is this will represent the superficial inguinal ring. Now this is how the extent of the inguinal canal can be mentioned with respect to these 2 rings, as a deep and the superficial inguinal ring. Now the thing is, now the deep inguinal ring, now again we'll look into the shape of that slightly in detail in the next slide. Now it is like now deep inguinal ring is said to be present between the deepermost part of the anterior abdominal wall, that is the fascia transversalis. Now this fascia transversalis, you might be again aware, deep to the transverse abdominis muscle there is a thin fascia, or deep to the transverse abdominis or inner aspect of the transverse abdominis muscle there is a fascia, thin fascia, that is called fascia transversalis. So the deep ring is an opening in the fascia transversalis. Likewise, now the superficial inguinal ring, which represents this one, this area. So this is nothing but a gap or opening in the aponeurosis of the external oblique muscle. So that is how the location can be mentioned, or extent of that one can be mentioned. Now we can make out from this diagram, so that it will be much more easier for you. We can make out here the anterior spine, the tubercle do not evident. So this is a inguinal ligament over here. We can make out the three muscles in order outside, the external oblique, internal oblique, transverse abdominis, and this is the fascia transversalis what you can make out over here. Now further inside, the fibrofatty tissue and the peritoneum can be seen as an innermost layer. Now what is important, relevant from this particular aspect or topic would be, in the fascia transversalis you see this opening. So this opening is a deep inguinal ring. So as you can make out over here, the canal passes like this obliquely, and this is the superficial inguinal ring, wherein you can see the content of the canal coming out through this opening, that is the superficial ring. So that is how the extent of the inguinal canal can be mentioned. Now, slightly in detail about what describing the shape related to the openings, two openings. Now, this is the, as we already seen earlier, this is the deep inguinal ring, which is said to be present or located or has a gap in the fascia transversalis, and location will be, how will you locate? Almost about approximately less than 1.5 centimeter, less than about say about 1.25 centimeter above the mid-inguinal point, about 1.5 or 1.25 centimeter above the mid-inguinal point. Mind you, mid-inguinal point. So mid-inguinal point is a midpoint between the anterior superior iliac spine and the pubic symphysis, between this and this. So this is a mid-inguinal point. So it is almost 1.25 centimeter above that. That is where the deep inguinal ring can be located exactly, and the shape wise it is said to be slightly oval in appearance. Whereas if you see the superficial inguinal ring, said to be, as we have seen earlier, so nothing but a gap in the aponeurosis of the external oblique muscle. So aponeurosis of external oblique muscle. So here the gap lies over here, just above and lateral. So location wise, just above and or immediately above and lateral to the pubic tubercle. So this represents the pubic tubercle over here. So this pubic tubercle just above that, this is the pyramidal shaped or a triangular shaped gap or opening that is seen in the aponeurosis of the external oblique muscle. So that is how the superficial and the deep inguinal rings can be located and appreciated. Now, apart from this, we have even looked into the boundaries related to the inguinal canal, so which has been systematically can be learned or remembered in this order as having to be the anterior wall. The canal is canal is having the anterior wall, posterior wall, roof, and the floor. So under these headings, we will look into that. Now the anterior. Now this is a cross-sectional view of the region of the inguinal canal. So this is now you imagine this to be the right-sided inguinal canal region. So the right-sided lower part of the anterior abdominal wall. So if you consider this to be the midline, so this is on the right aspect. So having to be seen, you can see the deep inguinal ring, superficial inguinal ring, and the passage is the inguinal canal. Now, what are the anterior, what are the boundaries or what are the structures will form the anterior wall? So this will be the anterior wall, and this is the posterior wall of the inguinal canal. And the roof and the floor will see the some other, we'll see in the other diagrammatic aspects. So they make up. Now, as of now, what is the anterior wall? So anterior wall, if you go in order from superficial to deep or outer to inside, it will be skin and superficial fascia, which is not shown over here. So it is assumed that skin and superficial fascia. After that, what you see, this line represents the external oblique muscle. So the outermost muscle, that's the external oblique muscle. After skin and superficial fascia, you see the external oblique muscle. Further deeper to that, you can see on the lateral 1/3 here. So that's what it mentioned. So as a lateral 1/3, this region, not the entire. This is the entire length of the anterior anterior wall of the inguinal canal. Only on the lateral 1/3 you see this muscle, that is the internal oblique muscle. So that is how the anterior wall. So anterior wall, so to be simple, form of skin, superficial fascia, then the external oblique muscle, and then the internal oblique muscle. That is how the anterior wall can be described. Whereas with respect to the posterior wall. Now this is the posterior wall of the inguinal canal. Now in the posterior wall, so what are the structures? Now the posterior wall, as you can make out over here, this is the this line represents the or denotes the fascia transversalis. So this will form one of the boundary to the posterior wall, fascia transversalis. Apart from that, now this is again magnified view. So we are now into the posterior wall, so which is formed by fascia transversalis. Now we'll first look into that, what are the structures in order, then we'll go back to the diagram. Now fascia transversalis in the whole extent, then conjoint tendon, then the inguinal ligament. Now you should be again aware, I hope you are aware now again. Suppose if I go back to this image, now the posterior wall, the anterior wall we have seen has to be the skin superficial fascia, external oblique and the internal oblique. Whereas with respect to posterior wall, this represents the posterior wall here. This is in order again. If I come from inside outwards, it would be fascia transversalis, the whole extent of the posterior wall, fascia transversalis. Medial part, medial 1/3 part would be the conjoint tendon, whereas the medial most part would be the inguinal ligament, part of the inguinal ligament. So these structures will represent the posterior wall. So that is how you can remember it as in the form of in an order with respect to the posterior wall. Now I hope you are aware of what exactly the conjoint tendon is. Nothing but the fusion of the aponeurosis of the internal oblique and the transverse abdominis. So to strengthen that the weakened area, potential weak space, that is how it will be helpful. So conjoint tendon. So that is how this will form the posterior wall. This represents the posterior wall of the inguinal canal. This is again one more surface view showing the same anterior structures forming the anterior wall and the posterior wall. I thought this is much more better. So this would be much more better and even to recollect in examination. So those are the anterior and posterior wall. Next, now again, this is just to make you understand, like just to make you aware, which you are already familiar. So this represents the muscle that is the internal oblique, and this represents the transverse abdominis. Now the aponeurosis of the internal and transverse abdominis in this region, so both of them where they fuse with each other forms a thickened tendinous pattern that is called as a conjoint tendon. So that will be one of the posterior wall boundary to the posterior boundary of the inguinal canal. Whereas now we will see the other boundaries, roof and the floor. Now this is again a sectional view which shows the inguinal canal with the content here. Now this will represent the roof of the inguinal canal. Now roof is by, as you can see, basically by these two muscles, that is the internal oblique, or you can see the lower arched fibers of internal oblique as well as the transverse abdominis muscle. So that will contribute. These two will contribute to the roof of the inguinal canal. Whereas the floor of the inguinal canal, as you can make out over here, so floor is nothing but the inturned lower free border of the aponeurosis of the external oblique muscle. So aponeurosis of the external oblique muscle has the free lower end. So that will enroll or return inside, giving rise to a passage. So that represents the floor of the inguinal canal. So that is with respect to these four boundaries with respect to the inguinal canal. This is again magnified view, so that we can see it in much more better way. So being the roof by these two muscles and floor by the aponeurosis of the aponeurosis of this external oblique muscle, which is nothing but inguinal ligament. So that will form the floor of the inguinal canal. Whereas now the contents of the canal, so that slightly differs in male and female. Now in male, the contents of the inguinal canal are considered to be primarily as a spermatic cord and the nerve, that is the inguinal nerve, which runs with the cord, whereas in females it is said to be the round ligament of the uterus along with the same nerve, that is the ilioinguinal nerve. So these are the primary contents of the inguinal canal. Now we look into the diagram here. Now this is a dissected specimen diagram. So as you can make out over here in this diagram, you can make out this represents the deep inguinal ring, and this will represent the superficial inguinal ring. Now the space or the space between these two, which has been shown as greenish appearance, so that represents the inguinal canal. So that is what the location of the inguinal canal and dissected specimen can be made out. You can make out the structure that is coming out through the superficial inguinal ring. As you can make out, this cord-like structure represents the spermatic cord. No doubt this is the male cadaver diagram. I mean the image from a male cadaver suggesting that this is the what suggests is this one, that is the spermatic cord being in communication with the blending with the testicular sac over there. So that is how, with respect to this image. Now the same image we have seen already how exactly the canal runs over here from the facial transversalis deep ring to the superficial ring, and the structure coming out as we consider, we can assume this one as being the spermatic cord, so representing the male inguinal canal. Whereas now this image shows as exactly the contents or difference between the male and the female differentiation of the contents in the inguinal canal. As we have seen earlier in the earlier image, you can see this one is the spermatic cord coming out and going in relation with the testis, whereas which is seen in as a content in the inguinal canal in a female anterior abdominal wall would be the round ligament of the uterus. Now this image gives in a better understanding what exactly the round ligament. As you can make out over here, uterus can be seen with the fallopian tubes on either side. So what it has been shown in blue color represents the ligament emerging or providing attachment to the uterus and to the lower aspect over here. So this represents the round ligament of the uterus. So this round ligament passes through as a passage in the inguinal canal, as in this canal. So passing through that one. So that represents the chief content in the as a content of the inguinal canal in females. That is the difference that lies between male and female inguinal canal as a content. Now this image again, just to get you a much more better understanding and in relation to the inguinal canal. As you can make out, this is the external oblique muscle. Once the external oblique is removed or reflected, this represents the internal oblique, and once this is reflected, you can make out the fibers from the transverse abdominis as well. After that, you can see the deepermost the fascia transversalis over here. So we can make out the deep ring as being a gap inside or gap in the fascia transversalis, and the superficial inguinal ring is best appreciated over here in this region, superficial, because superficial inguinal ring is seen as a gap in the aponeurosis of the external oblique muscle. So that is how we can in an order, so you can look into the structures which form the inguinal canal and its openings. There is a deep and a superficial inguinal ring. Whereas now a word about something called as the spermatic cord, so which we have seen already, as in the inguinal canal in males, this is the chief content, as being the chief content in inguinal canal in males, that is spermatic cord. Now this spermatic cord, cord-like structure, as the name itself suggests, as a cord-like structure which passes through the inguinal canal in males. Now this one, what exactly it is made up of, so that we should be aware what exactly it is made up of, or what exactly the contents of what exactly it consists of. So from that part of view, so we look into slight in detail about the spermatic cord, and mind you again, spermatic cord can be a 3-marker question, cross section, or the layers of the spermatic cord can be a 3-marker. So see to it that you be aware of this particular aspect as well in relation to the inguinal canal. Now, as you can make out from the diagram, this is the cross-sectional view of the spermatic cord. So what are the layers in order which will form the spermatic? To say the spermatic cord as a cord, so what is that which makes it a cord-like appearance? So now this image, from this image, before that, it basically has 3 layers. So if you go from outside inwards, if I go from outside inwards, so either way, either you can whichever which is convenient to you. So either inside outwards or outside inwards. Now if you go from outside inwards, so that outermost covering of the spermatic cord is by the external spermatic fascia, and the one slightly inner to that or middle would be the cremastic fascia, and the innermost layer that you can make out is considered to be the internal spermatic or inner spermatic fascia. So there is something called external spermatic and the internal spermatic fascia, and in between is the cremastic fascia along with the muscle of that, that is the cremastic muscle. Now, this layers, 3 layers, they are derived from, they are not, I mean, you have to even mention about from where exactly or what makes the, or what gives the appearance or what gives the, what forms, what helps the formation of the external or the internal spermatic fascia. As you can make out even from the diagram. So internal spermatic fascia, if you see the innermost, the internal spermatic fascia is considered to be a derivative of the fascia transversalis. So fascia transversalis is the innermost fascia which is seen on the inner aspect of the transverse abdominis muscle. So that will give rise to, or as a continuation from that one will be the internal spermatic fascia. Whereas cremastic fascia, so it is a derivative of internal oblique muscle. So we have already seen the second layer, the internal oblique muscle will contribute to the formation of the cremastic fascia, whereas the outermost layer, the external spermatic fascia, is said to be derived from the aponeurosis of the external oblique muscle. So that is how the 3 layers which are surrounding the spermatic cord are derived from 3 different aspects or 3 different parts related to the anterior abdominal wall. External spermatic fascia, just to again summarize, external spermatic fascia is derived from the aponeurosis of the external oblique muscle. So if I see into this diagram, now external spermatic fascia derived from the aponeurosis of the external oblique muscle. The cremastic fascia from the internal oblique muscle, derived from internal oblique, whereas the internal spermatic fascia is derived from the fascia transversalis, extension from the fascia transversalis. So those are the 3 basic layers that will give the cord-like appearance to the spermatic cord. Whereas now, after knowing these structures which form the cord, now we'll see what is exactly inside the cord. So now the things or the structures which are inside the cord, as you can make out from this diagram, we'll see from this diagram, will be so in a better way we can understand that. Now the chief content, like suppose if I say the chief content of inguinal canal in males is nothing but the spermatic cord, whereas in the same lines, if I say the chief content or the main content inside the spermatic cord would be the ductus deferens or the ductus deferens or the vas deferens. So as it is also called as vas deferens or the ductus deferens. That is the main content or the chief content of the spermatic cord. That has to be remembered. Along with that, you see surrounding that there are certain spaces that you can make out, irregular spaces. This represents a venous plexus around the ductus deferens. So which is particularly we use a term for the pampiniform plexus, which is seen around the ductus deferens. At the same time, so with respect to the ductus deferens, you can make out a vessel which is in close approximation to the ductus deferens, which is called as an artery to the ductus deferens. So obviously this is the source of blood supply to the ductus deferens. Apart from this as being the chief content inside the spermatic cord, apart from that you can make out on the like slightly in the posterior aspect, this would be there is one important, one of the larger arteries compared to other arteries over here. This is called the testicular artery. This is considered to be the branch from the abdominal aorta. So since spermatic cord goes in approximation or communication with the testis, so the artery, that is the testicular artery, is carried by the spermatic cord itself. So testicular artery, and likewise what we saw here, you can make out the irregular spaces. This represents again a venous plexus around the artery, which is again called as the pampiniform plexus. So we see pampiniform plexus of veins around the ductus deferens as well as around the testicular artery also. So these are the chief content. Apart from that, there is one more artery between the middle cremastic fascia and the internal that is this one, the internal spermatic fascia. What you can make out of the vessel would be the cremastic nerve and the cremastic vessel. So altogether, it has been said that it is said that the 3 set of arteries which has to be mentioned with respect to spermatic cord, as one being this one artery to the ductus deferens, testicular artery, and the cremastic vessels. So those are has to be mentioned as a content in the spermatic vessel from that point of view. Now other than that, you can make out here something called as related to the developmental aspect or during the descent to the testis. This is called as the remains or the remnant of the processus vaginalis. We will see what exactly is that the processus vaginalis in it. So these are the main contents which are to be mentioned with respect to the spermatic cord. Then one word would be, apart from these, you can even mention as the lymphatics. Lymphatics are also as considered to be as one of the contents which are seen as a content in the spermatic cord. So all these are, now this is how the text part which has been what we discussed right now with respect to the diagram. So all those things we can enumerate it as a content of the spermatic cord. So this can be again 3-marker question, mind you again. So see to it that you be aware of this particular aspect also. So the 3 arteries, what we saw, then the ductus deferens and the testicular artery, cremasteric artery, then artery with the ductus deferens which we already saw, then the venous plexus in the form of form, and the nerves, certain nerves as well. You can mention it as even the certain nerves, especially the genitofemoral nerve. So that also can be as mentioned as a content in the content in the spermatic cord. So those represents the finer details or inner details of the spermatic cord. Now after this, after knowing what exactly the inguinal canal is and where it is present in the lower part of the anterior abdominal wall, which is nothing but the intramuscular space. Now the space, when we say the space being narrow space to be present between the muscles of the anterior abdominal wall in the lower aspect, now that again also equally attached, with the potential weakness. So that also can give rise to certain pathological events can also occur through that potential weak space. So that is how the inguinal canal is. It is like from pathological point of view, it is considered a potential weak space in the lower part of the anterior abdominal wall. Now, wherein the contents of the abdomen can protrude through the anterior abdominal wall. So, but nature equally again simultaneously nature has provided us with additional reinforcement points or reinforcement features, so that the potential weak space, what we call it as pathologically, so this can be strengthened. Now, what are those factors or features that will strengthen the potential weak space rather than that? Instead of saying as potential weak space, that is further strengthened by certain features. Now, what are those features? One would be one aspect or one concept is called as the flap wall mechanism. Now, flap wall mechanism in the sense, now based upon the location of the or position of the inguinal canal, we said that it is an oblique space, intramuscular space. So that being present obliquely, what happens is whenever there is a raised intra-abdominal pressure, whatever the cause may be chronic cough which raises the intra-abdominal pressure, or maybe intra-abdominal mass, so that will always raise the intra-abdominal pressure. Now this canal being obliquely present, now the both anterior and posterior wall they will approximate each other with each other. So that gives so that is what I mean, they oppose with each other whenever there is a raised intra-abdominal pressure abnormal, not just a normal way, but whenever there is a raised abdominal pressure, intra-abdominal pressure abnormally, so that will lead to approximation of the walls. So that again acts as whatever which are inside the abdominal as a content will not protrude through the canal. So that is one concept. The second concept is called the guarding of the inguinal rings. Now, as you can make out over here, this is the superficial inguinal ring, and this is the deep inguinal ring. Now, they say, it is said that depending on the position and the structures around the rings, the superficial inguinal ring is more close approximated to the conjoint tendon, so that will guard this superficial inguinal ring. Likewise, and the deep inguinal ring is said to be guarded by the internal oblique muscle. So this being internal oblique muscle, so being very close approximation to the deep inguinal ring. So this will guard the deep, whereas the superficial inguinal ring is guarded by the conjoint tendon to some extent. Whereas the third concept is called a shutter mechanism. Here what happens is, again based on again related to the internal oblique muscle, This is external, this is internal oblique. It is said that internal oblique, like fiber one is if you closely visualize. Now the fibers will be like present in front of the canal, above the canal, behind the canal. So based upon the position of the fibers of the internal oblique being anterior, being covering the canal anteriorly, above and superiorly and posteriorly, whenever this muscle, the internal oblique contracts, so what happens is it will approximate the roof on the floor. So it will be like pulling the shutter upon the floor, that is floor which is formed by the inguinal ligament. So because of contraction of the internal oblique, it will shutter down, so that it will again prevent the contents of abdomen to be protruding through the inguinal canal. So that is the third concept. The fourth concept is also called the ball wall mechanism. Now here, why it is ball wall? Now what happens is cremastic fascia continues as further as the cremastic muscle, thin sheet of muscle, which is again a part of internal oblique muscle. If you remember, internal oblique muscle contributes to the cremastic fascia and the muscle simultaneously. Now we can see the this represents the brownish sheet that represents the cremastic muscle, which is surrounding the scrotal bag over here, along with the spermatic cord. So whenever the cremastic muscle contracts, it will pull the testicular sac towards the superficial inguinal ring. So that will occlude or block the superficial inguinal ring. So in that way, it acts as a wall, so that prevents again further prevents any of the contents to be entering through the inguinal canal, further entry going down into the leading to hernial condition. So these are the four concepts that are very much equally mentioned with respect to the inguinal canal. So that it's worth remembering again, being a potential space. Those were the four concepts that is worth remembering. Now, a word about one particular small topic in the inguinal canal would be something called as the inguinal triangle, or is also called by other name, one more name as the Hasselbalch's triangle. Now, Hasselbalch's or the inguinal triangle separately can be a 5-marker question. So 3 or 5-marker question has to be boundaries and contents has to be mentioned. So in that way, again, I suggest you be aware of this particular topic as well. Now in relation, where exactly or what exactly is the inguinal or the Hasselbalch's triangle? Now it is situated. Where do we mention it? It is situated in relation to the posterior wall of the inguinal canal. So this particular region is called this triangular. I can say triangular area is called as the Hasselbalch's triangle or the inguinal triangle. Now, what are the boundaries? It has a medial boundary, lateral boundary, and the base. So this being a triangle, and the apex lies over here. Now, the medial boundary, as you can make out here, medial boundary. Now, this muscle that you see on the background, this represents the rectus abdominis muscle. So, medial boundary is by the lower part of the lateral border of the rectus abdominis muscle. Or to be simple, you can mention it as medial boundary is by the lower part of the rectus abdominis muscle. So that is the medial boundary. So what is the lateral boundary of this triangle? It represents this artery, what you can make out as a branch emerging from external ilia. So this represents the inferior epigastric artery. This will form the lateral boundary to the Hasselbalch's triangle. What is the base of this triangle? Is contributed by the inguinal ligament. So this represents the inguinal ligament. So altogether, this will form the medial wall by rectus abdominis, laterally by inferior epigastric, and the base is by the inguinal ligament. Now there is something called as the floor of the Hasselbalch's triangle. Now floor is again contributed by you can say 2 parts. Now floor is said to be rather than 2 parts, or to be simple, the floor of this triangle is contributed mainly by a very thin sheet, that is the fascia transversalis, and further deeper to that, inner to that is the extra peritoneal tissue and even the peritoneum. That will form the floor of the inguinal or the Hasselbalch's triangle. So those represent the boundaries of the inguinal or the Hasselbalch's triangle. Now this is after going to these boundaries. Now what comes next would be the pathology related to that. That is the the hernias when we describe, so those are described with respect to this particular boundary, that is the inferior epigastric artery, which is nothing but one of the boundary to the lateral boundary to the SL-Box triangle. So keeping that in mind, so we will describe now the pathological aspect, that is the hernias. Now, as I said, now applied aspect related to this one. So what can be mentioned? How can you mention it? Now, one more thing in the same image. If I go back, if you look into this image, so which is this is the SL-Box triangle. Now, in the triangle, you see one dotted line running across here. Now, this dotted line from the umbilicus, this represents the umbilicus. From the umbilicus, you see this dotted line. Now, this dotted line represents the obliterated umbilical artery. So, obliterated umbilical artery, once after birth of the child, gets obliterated, and it will be present henceforth as a medial umbilical ligament. So, this represents the medial umbilical ligament. Now, this medial umbilical ligament further divides the triangle into two parts, into medial half and the lateral part. So, medial part, so same way, the medial part of the triangle is the stenton. Now, see, if you consider this, I mean this as the obliterated umbilical artery. Now, this is the medial part of the triangle, and this is the lateral part of the triangle. Now, medial part of the triangle is strengthened, or it is reinforced by this conjoint tendon, as you can make out here. So, this represents the conjoint tendon. The medial part of the triangle is reinforced by the conjoint tendon, but the lateral part of the triangle is said to be quite weaker, in the sense it is covered only by the fascia transversalis and the extraperitoneal tissue over here. So that is how we divide. That is how the umbilical, that is obliterated umbilical artery in the form of medial umbilical ligament divides the triangle further into medial and lateral half. So medial part, remember, it is slightly stronger. You can say that the lateral part is slightly said to be comparatively slightly a weaker area. So hence, since lateral part being a weaker area, so it is more vulnerable for the protrusion or the herniation to occur. So hernia more commonly occur from this particular region, so especially the direct. Now we'll see what exactly are the terms that we are supposed to see as a pathology related to the inguinal canal in the form of hernias. Now, before that, defining the hernia. So I think I assume that you already very much aware of what is hernia. Any abnormal protrusion or any protrusion from any particular region in any particular part is called as a hernia. So that is abnormal protrusion. Now this hernia, which is with respect to inguinal canal, are called as the inguinal hernias. Now this is again further divided into two types, as a direct inguinal hernia and indirect inguinal hernia. Now how do we, or when do we call it as a direct and indirect? That is the question now. Now, if you take a first the direct inguinal hernia, direct is direct because it directly protrudes the posterior wall of the inguinal canal. The posterior wall of the inguinal canal. That's what is called the direct inguinal hernia. So it will direct is direct because it will directly protrude. The contents protrude through the posterior wall of the inguinal canal. Remember, posterior wall of the inguinal canal will be directly protruded by the contents of the abdomen, and those contents, they make a direct entry or appearance in the inguinal canal. So by passing or protruding through the posterior wall of the inguinal canal. Now what is the posterior wall of the inguinal canal? You might be aware again, so which we have already seen, the posterior wall of the inguinal canal. Just to again make you again recollect that. Now this is the posterior wall of the inguinal canal, which was formed by fascia transversalis and the conjoint tendon and contributed by the inguinal ligament. So that was. Now any abdominal content protruding directly through the posterior wall and making an appearance into the canal, so that is called as a direct inguinal hernia. That is how you describe or you define the direct inguinal hernias. Now one more thing along with that is what you have to always remember is now you have to always mention the hernias, direct or indirect, with respect to even as an important landmark as an inferior epigastric artery. So now direct inguinal hernia is direct when it protrudes through the posterior wall of the inguinal canal. But where on the posterior wall of the inguinal canal, it should be like medial when the hernial contents protrude the posterior wall of the inguinal canal, by in such a way that staying medial to the inferior epigastric artery. This is the inferior epigastric artery, this is the medial part, medial aspect of inferior, this is lateral to the inferior epigastric. So any abdominal contents protruding through the directly through the posterior wall of the inguinal canal, staying medial to the inferior epigastric artery, they are called as direct inguinal hernias. So what is direct inguinal hernias? There is one more called indirect, we'll see that later. Now direct inguinal hernias are said to be more common in elderly people because of laxity of the abdominal muscles. So that is how it is very more common in older people. Whereas indirect, now indirect, it is the one that will enter into the inguinal canal. The hernial contents, they will enter into the inguinal canal by passing lateral to the inferior epigastric artery. Remember, now if you consider in this diagram, there will be a superficial inguinal ring over here, and the deep inguinal ring will be over here. Now the abdominal contents pass through the deep inguinal ring, that is lateral to the inferior epigastric artery, and then they make an entry into the inguinal canal. So that is how we describe the indirect inguinal hernia. So that is the difference between direct and indirect. Direct, they don't pass through any of the opening, be it superficial or they are not related to the ring, deep ring, but instead they pass directly through the posterior wall of the inguinal canal, being medial to the inferior epigastric artery. Whereas indirect is they pass through they enter through the deep inguinal ring, being staying lateral to the inferior epigastric artery and entering into the canal. So that is how you can differentiate the direct and the indirect. So to be simple, direct is will stay medial to the inferior epigastric, and indirect hernias are nothing but more lateral to the inferior epigastric artery. So inferior epigastric artery forms a very important landmark over there. Now the indirect inguinal hernias are unlike direct. We said that it is more common in elderly people, whereas indirect are more common in children and even the younger adult, and that is more particularly related to one of the developmental remnant as a process vaginalis. We'll see that what exactly is that. So that better understanding. Now these are again inguinal, indirect are more common in indirect are more compared to direct and indirect. Indirect are more commonly occur, and no doubt these occur more commonly in males rather than compared to the female. So that is how we can describe that. Whereas further for better understanding, the indirect inguinal hernias are of two types. One is as a congenital and the other one is acquired. Now congenital is again, as I said earlier, it is related to the processus vaginalis. Now what exactly is the processus vaginalis? Now this, if you will get it, maybe you might recollect if you have gone through the embryology part. Now we are looking at this image again. Now as you can make out, the testis initially it will be slightly in the higher up. Now it has to descend to the scrotal region. Now for the testis to descend into the scrotal region, it needs some guidance. That guidance is based upon the projection of the peritoneal sac over here. So this peritoneal sac, as you can make out here, it is projecting down. So it is projecting down towards the scrotal sac. Now that projection of the peritoneal sac as a guide for the descent of the testis is called as processus vaginalis. Now as the testis, as you can make out here, testis is descending down along with that. So as the testis enters into the exact location, that is in the scrotal sac, now the process, you can see the process vaginalis is a narrowing and gets obliterated. Only now it is present as a sac around the testis, which is called as the tunica vaginalis in an adult life. So this is the one, but rest of it gets obliterated. If the processus vaginalis remains patent, so that will give rise to the congenital indirect inguinal hernia. Now this is how. Now this is the sac. Now you can see the obliterated the process vaginalis obliterated. Now this is the tunica vaginalis. It's a sac which is present after the descent of the testis. So that is how we can describe. These are the again structure to show, just to show the how exactly or where exactly this tunica vaginalis is present. So with respect to that, now testis again, that is a different story altogether. So we will get to know later. So that is with respect to the congenital. Now acquired, as the name itself suggests, more commonly due to chronic raised intra-abdominal pressure, be it in the form of weightlifting or a chronic cough. So all those things can give rise to protrusion of the abdominal contents. So that is how the acquired inguinal hernias the cause for the acquired inguinal hernias. So now again a word about the difference between direct and indirect, as depending on what what are the coverings of that particular sac. When we say abdominal contents are protruding through the inguinal canal, so it is obviously covered by certain layers. So those layers has a slight difference between the as a covering from covering to difference between direct to indirect. Now if you see first the coverings, what can be made out with respect to the, as you can see, these are the contents of terminal protruding through the canal, giving rise to this the herniation. Now the contents would be like if I go from inside outwards, peritoneum. So as you can make out here along with the content, you see this has been labeled over here as in red color, so represents the peritoneum. Further outer to that is extra peritoneal tissue, not marked but otherwise after the peritoneum, so extra peritoneal tissue, and further outside to that you can see this is the internal spermatic fascia, and further outer to that cremastic, and then the external spermatic fascia, and then the skin. So that is how the contents of the indirect inguinal hernia as the coverings of the indirect sac, that is the indirect hernial sac. Whereas if you compare it with the direct one, so more or less same, slight difference would be peritoneum. Same, you saw peritoneum there, and you are seeing peritoneum here as well. In the direct, you saw extra peritoneal tissue in the indirect, and there is extra peritoneal tissue in the direct as well. What the difference, what we don't see more between these two would be internal spermatic fascia in the indirect, but we don't see. But instead of that, we see fascia transversalis. Now why? Because now direct enters, makes an entry, piercing, projecting through the wall, posterior wall of the inguinal canal. That's why there is we can make out the fascia transversalis. But in the indirect, it passes through the deep ring. So that's the reason why we don't mention it as, we don't see the fascia transversalis in indirect, but instead we see it as an internal spermatic fascia. That is the only difference. Other than that, more or less after that, the cremastic muscle, external spermatic fascia, and the skin almost remain same. Now what further? Just like the indirect inguinal hernia, which was divided into the congenital and acquired. Same way again, direct inguinal hernia is also are also divided as a medial direct and lateral direct, depending on the direction they move across. Now, medial direct, when do we call it as medial direct? So when the hernial sac or contents are covered by the conjoint tendon, and the same direct inguinal hernia can also be called as lateral direct when they are not covered by conjoint, but instead they are covered by the cremastic muscle. So that the difference, based upon observing those difference, you can describe. Now even in the image, now as you can make out here, now like medial direct and lateral direct. Now from this image, now direct hernias, they have to be medial to the inferior epigastric artery. So direct inguinal hernia can be medial direct or lateral direct. Now how medial direct? If the direct inguinal hernia is medial direct, now this we know that this is the background, what you say is the conjoint tendon. Now if the direct inguinal hernia, so see, direct inguinal hernia is when the herniating contents are related medial to the inferior epigastric artery. So direct inguinal hernia can protrude through the entire posterior wall here. Now if the direct inguinal hernia is arising from here or protruding through this wall, that is this part of the posterior wall of the inguinal canal, in relation to the conjoint tendon, it is called direct medial, because it is in approximation with the conjoint tendon. Same way, direct inguinal hernia is said to be lateral direct. So when it is away from here, but it is when it is not covered by the conjoint tendon. So when the hernia, direct inguinal hernia, is projecting again through the posterior wall of the inguinal canal, but away from the conjoint tendon. So this would be called as the direct or lateral direct inguinal hernia. Medial direct inguinal hernia, lateral direct inguinal hernia. That is how you can describe or classify the direct hernias as well. So that is how the topic about the topic today, that is inguinal canal. What we saw the extent and direction, we saw the boundaries, the contents, and also we even saw with respect to one particular aspect with respect to that inguinal triangle, and also the other aspect as being the hernias. So that will complete today's topic, and since we have 10 more minutes, we'll run through some of the MCQs that might be relevant to you from examination point of view. First MCQ, as you can make out over here. Now, the question can be inguinal canal is what? Now if you go through that, so it is again what exactly is inguinal canal? So which can be the most appropriate answer for to describe the inguinal canal? So it is a horizontal. Definitely it is not horizontal. It is present over the lateral one third. Definitely it is not. It is absent in females. Inguinal canal definitely not. And would be the more appropriate would be so this one. So the option B is considered to be the more appropriate choice for the this particular question. So that is how the MCQ. Now whereas the second MCQ with respect to this one, the deep inguinal ring, what exactly is a deep inguinal ring? So either how can you like which is more relevant to this particular? Is it an oval shaped gap in external oblique? Definitely not. So if it is present above the lateral to the tubercle, definitely not again. Whereas superficial and deep both are they both one the same? Not. So the option for this particular MCQ would be none of the above. So obviously it won't fit any of the options for this particular aspect topic. Whereas third, contents as a contents of inguinal canal. So what are exactly the contents of inguinal canal? So is it what is the better option over here? Is it a fallopian tube or is it a ureter in case of males? Definitely not. And is it a round ligament of the uterus in females and spermatic cord in males? Or is it all of the above? So obviously this fits into as a better option for this particular question. Whereas the fourth question would be factors which maintain the integrity of the inguinal canal, which we have already seen the concepts of the 4 or 5 concepts, 3 or 4 concepts. One was this one and this one and this one. So all are true of this one. So what option fits for this question would be none of the other. So because all of them are true except the fourth option. Whereas fifth one, Hasselbalch's triangle. For Hasselbalch's triangle, boundaries all of them are correct except inferiorly by lateral of the inguinal ligament, laterally by inferior epigastric artery, definitely, medially by lower lateral border of rectus abdominis muscle, floor is by the peritoneum along with the fascia transversalis. So obviously these three are correct. So what which is not is this one, that is the inferiorly, because inferiorly is by the medial half of the inguinal ligament rather than lateral half of the inguinal ligament. The sixth one, common symptoms of inguinal hernia when the patient presents would be all except presence of lump, or is it dragging and pain, or option both, or is it correct, or is it severe vomiting associated with sweating. So what are all which are correct? So definitely this is not the one which is correct. These are very common presenting symptoms. Now the one more MCQ, all are correct related to coverings of the spermatic cord. Now we recollect spermatic cord covering internal spermatic fascia derived from what? As a fascia transversalis, premastic fascia derived from internal oblique, external spermatic again derived from external oblique. Whereas what is not is premastic fascia being derived from as a content, as a transverse abdominis muscle. So that is the seventh one, and should be the last, I suppose. Regarding the inguinal hernias, all are correct. As inguinal hernias, not seen in females. Indirect inguinal hernias are more common in children, definitely correct. Indirect hernias are more common compared to the direct. Comparison between direct and indirect. Femoral hernias are more common than inguinal in females. That is also correct. But what is not is this one. Inguinal hernia doesn't mean that inguinal hernias is, females are not immune to inguinal hernias. That is not a correct statement. So inguinal hernias are also seen in females, but very less frequent. So that is our last, but that should be last one. So which is correct? No, inguinal, indirect inguinal hernial sac does the canal by passing lateral to the inferior, or direct passes lateral to inferior epigastric artery. Both A and B are correct. Or femoral hernias are more common in males. So which is most correct? Correct would be this one, as being this statement as indirect or more lateral states lateral to the. That is about today's topic for now. So thank you very much. If you have any doubts, please let me know. Thank you very much.
Good afternoon to all. Today we are going to do a reflexes. This is a part of a motor system where we are going to do reflexes. In the reflexes there are two types of, it is classified into two types. One is superficial reflex, next is deeper reflex. In superficial reflexes we have corneal reflex, conjunctival reflex, palatal reflex, abdominal reflex and plantar reflex. Corneal reflex, conjunctival, palatal reflex, this we are going to do in cranial nerve examination. I will just tell the root values for afferent and efferent for this reflex. All the other we are going to demonstrate today. For corneal reflex, afferent is trigeminal nerve and efferent is seventh nerve, that is facial nerve. Conjunctival reflex, afferent is trigeminal and efferent is again the facial nerve. Palatal reflex, afferent is ninth cranial nerve, that is glossopharyngeal nerve. Efferent is 10th nerve, that is vagus nerve. These three reflexes will cover in the cranial nerve examination in the coming practicals. Today we will do abdominal reflex and plantar reflex in superficial reflexes. For abdominal reflex, this is how you should examine. You should take a blunt object and stroke from outer border towards the umbilicus on 6 sides, from above to down, then in the middle from lateral to medial side, from down from lateral to medial side. Here you see a contraction of a paraspinal muscles. This for a abdominal reflex with a root value of T7 to T12. Now we will see the plantar reflex which we are going to demonstrate. Okay. So, for plantar, in superficial, as I told, it is abdominal reflex. You saw in the slide. Now we will do a plantar reflex. Plantar reflex we have to do in the lying down position. You should instruct the subject, and as we told in the previous experiment, we should stand on the right side of the subject. Before we do, you should use a sanitizer. Expose the area which you want to examine. Plantar reflex, this is the superficial reflex with a root value of L5, S1 and S2. For this you should fix the ankle joint. Okay, you should fix a ankle joint, then take a key. Blunt object, here I am taking a key. Okay, with the key you stroke from lateral side to the medial side. You will see the flexion of all the toes. Same thing you should repeat in the other limb. You should stroke from lateral to the medial side where you see a flexion of all the toes. I will show. Again, you should fix the ankle joint, then stroke from lateral side towards the thumb. So you will see a response as flexion of a toes. You should repeat the same thing on the other sole by stroking from lateral to the medial side. It's a contraction of a flexion of a toes. This is about a plantar reflex. Then coming to deep reflexes. In deep reflexes, first is jaw jerk, where the sensory and motor part of a trigeminal which takes part in the jaw jerk. Second, biceps, C5 and C6 are the root values. Triceps, C6 and C7. Supinator, C5 and C6. These are the upper limb reflexes. Jaw jerk, biceps jerk, triceps and supinator. Then in the lower limb, knee jerk, that is L2, L3 and L4, ankle jerk S1 and S2. In this, first I am going to demonstrate jaw jerk. For jaw jerk, you should keep your thumb over a chin, then hit with the smaller end of a knee hammer over the thumb. Then there is a slight closure of a mouth is a reflex for a jaw jerk. I repeat, keep your thumb over a chin, then hit with the smaller end of a knee hammer. Then the response is slight closure of a mouth. This is regarding a jaw jerk. Then coming to the upper limb one, that is biceps. You should ask the subject to relax, loose breath, full loose breath. Ask the subject to relax and keep over your hand and ask him to put weight over your hand. Then it should be loose enough. Then keep your thumb over the insertion of a biceps tendon. Then the area which the muscle which you want to examine should be exposed. So this is a biceps. When you hit over the tendon, then you will get a contraction of a biceps with a flexion of a forearm. I repeat, when you hit over the tendon, then you will get a contraction of a biceps and flexion of the forearm. Same thing you should repeat on the other hand. You should ask the subject to place over your hand, then place a thumb over the insertion of a loose insertion of a tendon, then hit over it, then you will see the contraction with a flexion of a forearm. This is for a biceps. Then for a triceps, in the upper limb, biceps is over. After that, we will see a supinator. Supinator you should hit over a insertion of a supinator where you see a contraction of a supinator, loose contraction of a supinator muscle. Same thing you should repeat for a loose for the other hand. Then you should see the contraction of a supinator muscle. This is for a supinator. Then coming to the triceps. For a triceps, you keep a hand over here, then ask the subject to dangle his hand over your hand, then ask him to put a weight over the hand, loose breath, full, relax it. Hit it over the insertion of a triceps. So you will see a extension of a hand with a contraction of a triceps muscle. All this experiment you should do in sitting and a lying down position. Now I am doing a sitting position of a triceps jerk. Loose breath, full parabolic. Hit over it, so you will get a extension with a contraction of a triceps muscle. Okay. So this is for a biceps, supinator and triceps jerk in the both the upper limbs you should do. Then coming to the lower limbs. In lower limbs we have two reflexes. One is knee jerk, second is ankle jerk. For a knee jerk you can do either by dangling the lower limb or by crossing the limbs. Here I do in the dangling and both also on the crossing the limbs. So you should hit over the insertion of a muscle here. So there is a extension of a limb when you hit over a insertion of a muscle. Then coming to other limb, same thing, there is a extension of a lower limb with a contraction of a quadriceps muscles. Then for a ankle joint, ತಿರುಗಿ ಮಂಡಿ ಒಂದು ಕಾಲಿದೆ ಮುಂದೆ ಇನ್ನಿಂದ ಈ ಕಡೆ ತಿರುಗಿ ರೋಡ್ ಕಡೆ ತಿರುಗಿ ರೋಡ್ ಕಡೆ ತಿರುಗಿ ಆ ಕಡೆ ತಿರುಗಿ ಒಂದು ಕಾಲಿದೆ ಸರ್. ಸೊ ಫಾರ್ ankle jerk you should eat over the insertion of a gastrocnemius. Before doing a reflex you should extend the ankle, then hit over a attachment of a tendon Achilles. So you will see a extinct flexion of a limb with contraction of a soleus muscle. ಈ ಕಾಲು ಕೆಳಗಿರಿ ಆ ಕಾಲು. Same thing you should repeat on the other leg. ಇನ್ನಿಂದ ಬರಬೇಕು. Dorsiflex with a hitting over the tendon Achilles. So you will get a contraction and a extension of a soleus muscle with the extension of the ankle. This is for a lower limb reflex. In lower limb we did both. One is ankle jerk, second is a knee jerk. This is in a sitting position, upper limb and a lower limb reflexes. Now we will see here in lying down position the same reflexes will be demonstrated by Shilpa ma'am. Okay.
Before starting any examination, make sure that you clean your hands. Examination should be done with clean hands or with the gloves on. Yes, so till now we have learned what are the different tendon reflexes. We have superficial reflex and the deep tendon reflexes. Superficial reflexes are polysynaptic reflexes, whereas deep tendon reflexes are monosynaptic reflexes. These monosynaptic reflex, as you know, you have a receptor. Here the receptor is in the form of muscle spindle. So whenever you stretch this muscle spindle, there is reflex contraction of the muscle. So this is what we are going to elicit by eliciting the deep tendon reflexes. That is the stretch reflex of the specific muscles. So we will till now you have learned how to examine these reflexes in sitting position. Suppose you have a patient, instead of a subject you have a patient who cannot sit, who is bedridden. Then you have to do the examination in supine position. So how to do the examination in supine position we will demonstrate now. For the biceps tendon reflex, you should make sure that the person puts his forearm in the flexed position on his chest or the abdomen. It should be semi-flexed. You have to place your thumb over the biceps tendon. Make sure that your thumb is over the biceps tendon. You have to hold the knee hammer such that you dangle it. You have to just give a hit through your wrist joint. You should not use your elbow in order to flex. You have to use your wrist joint to give the tap. Use the narrow end of the knee hammer and give a tap on the biceps tendon. You see the contraction of biceps muscle and also flexion of the forearm. Similarly, on the other limb, you have to hold the thumb on the biceps tendon and give a hit on your thumb. You should observe for contraction of biceps muscle and also the flexion of the forearm. Then for the triceps jerk, we will do the demonstration on the same limb. Again, flex the forearm, rest the forearm on the chest. With the broader end, you have to hit over the insertion of the triceps muscle. Ask the person to stay calm and keep the limbs loose. You see the extension of the forearm and the contraction of triceps muscle. Similarly, you have to examine on the other side. Loose. You have to hit over the insertion of the triceps muscle. Look for extension of the forearm and contraction of triceps muscle. For supinator reflex, yes, the root values of biceps reflexes, biceps deep tendon reflexes C5, C6. For the triceps is C6 and C7. For the supinator jerk, it is easier to hold the hand in a handshake position, so that you hold the hand in a comfortable way, and you have to hit over the insertion of the supinator muscle. Look for supination of the forearm. Similarly, support the forearm and then give a tap on the insertion of supinator muscle. You can also support it this way. You can hold the forearm and the wrist in a flexed position, or you can hold it in a handshake position. You look for supination of the forearm. So this is about the deep tendon reflexes in the upper limb. The root value for supinator jerk is again C5 and C6. Coming to the knee jerk. Raise the clothes above the level of knee. Put your hand below the knee. You can take the support of the other knee and make sure that the person leaves all his strength on your arm, and the person should leave the limb loose, otherwise you won't get the reflexes. You can use the broader end or the narrow end. Palpate the insertion of the quadriceps muscle and then hit over the quadriceps tendon. You get the extension of the limb with contraction of the muscle. It is better you lift the clothing above the level, so that you can also observe the contraction of quadriceps muscle, at the same time extension of the lower limb. Similarly, you take the support of the person's other limb. You have to hold the limb that you are going to elicit the tendon reflex. Make sure that the person leaves the limb loose and then hit over the tendon. Look for extension and contraction of the quadriceps muscle. Then the root value for knee jerk is L2, L3 and L4. L2, L3 and L4. Next, coming to the ankle jerk. In the ankle jerk, in supine position, how to elicit ankle jerk? Make the subject keep the limb on the shin of the other limb. The knee joint should be semi-flexed and there should be slight extension of the ankle joint. Hold the great toe, make slight extension, and you have to hit over the Achilles tendon. Ask the person to leave the limb loose. With the broader surface of the knee hammer, you hit over the Achilles tendon. If you look carefully, you can see for the contraction of soleus muscle and plantar flexion of the foot. Similarly, elicit the plantar reflex over the other limb. Slight extension of the ankle joint, flexion at the knee, slight extension of the ankle joint, you hit over the Achilles tendon. So this is how you elicit for plantar reflex. Sorry, ankle jerk. Ankle jerk, the root value is S1 and S2. Then you have also observed how to elicit plantar reflex, which is a superficial reflex in lying down position. Suppose this is the foot, this is the medial end of the foot and this is the lateral end of the foot. You have to give a stroke from the lateral end towards the medial end. I repeat, You have to give a stroke from the lateral end towards the medial end. Look for flexion of the toes. Suppose if there is any abnormality like upper motor neuron type of lesion, then you see for the upgoing plantar, that is the great toe will go for dorsiflexion. Great toe dorsiflexion, at the same time fanning out of the other toes. So you do a stroke on the foot. I am demonstrating this on the foot. So considering this as a foot, you have to give a stroke from lateral to medial end where there is great toe extension and fanning out of the other toes. So this is how you have to do it in the foot. You can use a key or you can use a sharp end of the knee hammer. You can use the sharp end of the knee hammer, stabilize the ankle joint, then from the lateral give a stroke from lateral to medial. Normally we see flexion of the toes. So this is about the demonstration of different deep tendon reflexes and superficial reflex, that is the plantar reflex. So we have demonstrated how to examine in the upper limb and in the lower limb. Suppose many students, you might have not taken the knee hammer at home. You may not be having knee hammer at home. Then how to elicit these reflexes, how to practice these reflexes? You can use any object which is not harmful. Make sure it should not be harmful, it should not break the tendon of the subject. It should be a blunt object which is heavy at the end. It should be a blunt object which should be heavy at the end, and it should be lightweight. The handle should be lightweight. You can also use a stethoscope. If you have taken stethoscope, suppose if you have taken stethoscope at home, you can use the bell of the stethoscope. I will demonstrate to you how to do the examination using the bell. Biceps tendon reflex, you can use the bell, hold it like this. This should dangle, the bell should dangle. You have to hold near the end, then give a stroke on your thumb. Give a stroke on the muzzle. Got it? You can give a stroke with the help of the bell. Similarly, knee jerk. So whenever you are eliciting knee jerk, make sure that the limbs should be left loose and you have to support the limb on your upper limb. So this is how you demonstrate for knee jerk when you don't have your knee hammer. Yes, you can use the bell or any blunt object which has a handle, and the handle should be lightweight. You can use it. Whenever you are giving a stroke, make sure that your stroke should come from the wrist. Your movement should be at the wrist. Don't hit from the elbow. It should be free. The movement should be free and it should give a good stroke when it falls over the tendon. So this is how to elicit the deep tendon reflexes and superficial reflexes. Very easy and simple to remember the root values. See, for the ankle jerk, 1 and 2, yes, 1 and 2, 2, 3, 4. I repeat, 1, 2, yes, 1, 2, 2, 3, 4, L, 2, 3, 4, 5, 6, 5, 6, C5, C6, C5, C6, and the triceps C6, C7. So this is how you can easily remember the root values. I repeat, S1, S2, that is 1, 2, S, 1, 2, L2, 3, 2, 3, 4, 1, 2, 2, 3, 4, 5, 6, 5, 6, 6, 7. S1, S2, L2, L3, L4, C5, C6, C5, C6, C6, C7. So this is how you can easily remember the root values of different deep tendon reflexes. Remember, all the deep tendon reflexes are monosynaptic reflexes. That means it has an afferent limb, it has a center, and it has a efferent limb. The afferent limb is in the form of sensory nerves, sensory nerve 1A or 2. and the efferent limb is the alpha motor neuron. The center is these spinal cord regions what we have numbered, S1, S2, L1, 2, 3. They are all the centers where this deep tendon reflex is going to get integrated. So all these deep tendon reflexes are monosynaptic reflexes, whereas superficial reflexes are polysynaptic reflexes. So this is about the examination of reflexes. In exams, they will ask you to elicit the deep tendon reflexes, to number the root values, to elicit the reflexes in supine position as well as in sitting position. Then questions may be asked based on the theory, like what are the types of reflexes, what are the characteristics of reflexes, properties of reflexes. They may also ask you about the upper motor neuron lesion and lower motor neuron lesion. So this is about this practical class, that is eliciting examination of reflexes. Thank you. So good afternoon. Till now you have observed how to elicit various reflexes, for example the superficial reflexes and the deep reflexes. So in the superficial reflexes we have demonstrated how to examine, means how to elicit the plantar reflex. In that even the abdominal reflex is also important, which we cannot show it here. Okay, usually it is given in the text. You have observed there is a figure, means showing different quadrants of the abdomen, and the abdomen is being divided into 3 portions. Above the level of the umbilicus, that is supraumbilical area, umbilical area and the infraumbilical area. So in each area the stroke will be different. Okay, usually it will be delivered from lateral towards the medial side, that is like this in the upper quadrant, and in the middle quadrant it is from lateral towards the umbilicus, and in the infraumbilical area it is from downwards, from lateral to the medial side. This is again a superficial reflex. Okay, the same thing is shown here. So look at this. This is the abdominal reflex, which is a type of superficial reflex. Okay, so all superficial reflexes, they are polysynaptic. Okay, and in this, if you observe carefully, the stroke, the direction of the stroke is different for the supraumbilical area, for the umbilical area and for the infraumbilical area. So this is along the lower costal margin. So you are delivering the stroke from lateral to the medial side. Okay, so in the same way you have to repeat it on the other side. Next, in the umbilical area, this is again from the lateral towards the umbilicus. On either side you have to do. Then in the infraumbilical area, this is from lateral to the medial side. Okay, so what is the response you get when you strike or when you stroke the skin over the abdomen? So what will happen? There will be contraction of the anterior abdominal wall because of the contraction of the muscles which are involved in the forming the anterior abdominal wall. So this is a superficial reflex. The next was the very important reflex, most commonly asked, theoretically also it is very important, that is the plantar reflex that they have shown. Okay, you have to remember the root value. The plantar reflex, the root value is L5S1, and as the madam has shown you, take this as a foot and this is your great toe. Okay, so the thumb will become your great toe and all other fingers will become the toes. So what you are supposed to do is you have to take a key. Since you don't have the knee hammer at your place, you can use the key, any key you can use, and use the blunt end of the key, and you have to start striking the skin from the base of the heel. Okay, from the heel, strike along the lateral border, then go along the, then turn medially, and you have to stop near the base of the great toe. Okay, I'll repeat. So you have to start striking the skin from the lateral border of the foot, that is from the base of the heel, go along the lateral border, turn medially to reach the base of the great toe. So what response do you expect? As as you all know, this is the ankle joint. I am telling this is we are comparing the hand to a foot, and this will become, the wrist joint will become the ankle joint. So which are the movements which occur at the ankle joint? It is the, which is this movement? Plantar flexion, and this is dorsiflexion. Okay, so see here, this is the plantar flexion and this is the dorsiflexion. In a normal person, when you strike the skin over the foot, what response do you get? First, there will be flexion of the, there will be flexion of the ankle joint. Okay, so first there will be flexion of the ankle joint, that is plantar flexion. All the toes including the fingers will bend. Okay, so that is the first response. Then there is flexion at the interphalangeal joints also, and there will be bending of the great toe. All the other fingers will also flex. Look at, they are coming closer. All the fingers, they are coming closer. That means there will be adduction of the fingers. So these are the components of the response what you get for a plantar reflex. I repeat. So what is the response you get in plantar reflex? There will be flexion, plantar flexion at the ankle joint. Then there will be flexion of the toe along with the flexion of the fingers because of flexion at the interphalangeal joints. Then there will be adduction of all the fingers. The fingers are coming closer, they are not spreading apart. Okay, so the fingers are coming closer. So there will be adduction of all the fingers. So this is a normal response. When you get such a response, then it is called Babinski sign negative. Okay, so the Babinski sign negative, it is seen in normal person, but when it becomes positive, it is an indication of upper motor neuron lesion. Okay, so that is why the plantar reflex is very important. So what is Babinski positive? In Babinski sign positive, what will happen if the person is suffering from upper motor neuron lesion? So what happens? Instead of plantar flexion, there will be dorsiflexion. Okay, so there will be dorsiflexion and there will be extension of the great toe and also extension of all the fingers. Look at the fingers, they will span out. All the fingers will span out. There will be abduction of all the fingers. Okay, and then it is called the Babinski sign positive, which is clearly suggestive of upper motor neuron lesion. By now the term upper motor neuron and lower motor neuron terms, it is familiar to you. So anything above the level of anterior horn cells which are located in the anterior horn of the spinal cord, these neurons will constitute the upper motor neurons. Okay, that is above the level of anterior horn cells, which may include the axonal fibers which carry the various sensations. Then they make synaptic connections with the various nuclei which is present in the brainstem. Go to the thalamus, then to the motor cortex itself. Okay, so all these neurons, they form the upper motor neuron. Then anything below the anterior horn cells will form the lower motor neuron. So this Babinski sign, it will help us to differentiate whether the person is suffering from UMN or LMN lesion. Okay, so always the plantar reflex has to be elicited in the supine position. Fine, and any reflex, if you want to observe the response, if you want to get a good response, positioning of the limb is very, very important. You have observed how the teachers, they used to fix the limb. So that is very, very important because you have to relax the joint. Okay, only then you will get a response. Suppose if the person is very anxious and if he becomes conscious, okay, you will never get the response. So in that case, is there any alternative to get the response? Yes, we do have a manual which needs to be followed in order to elicit the response for the particular type of reflex. So that is Gendrasic's manual. Okay, so that is the Gendrasic's manual. So what is this Gendrasic's manual? So as you all are aware, most of the deep jerks, they are monosynaptic. The receptor is the muscle spindle. You have learned in detail about the structure of the muscle spindle. Okay, the afferent usually it is A alpha fiber, and efferent you have the gamma motor neurons, isn't it? So if and these muscle spindle, it is very much sensitive to gamma motor neuron discharge. Okay, and these gamma motor neurons, they are under the influence of higher centers that you have already learned, the cerebral cortex, the basal ganglia, the cerebellum, and some other structures, they also influence the gamma motor neuronal discharge. So by increasing the gamma motor neuronal discharge, you can actually exaggerate the response obtained from the deep reflexes. So how will you increase the gamma motor neuron discharge? It is by the Gendrasix maneuver. So what is this Gendrasix maneuver? Usually you ask the subject to hook his fingers. Okay, usually ask the subject to hook his fingers to get a response in the lower limb, or clench his jaw. Okay, ask him to clench his jaw and hold it tightly for some time till you examine for the reflexes. By doing these maneuvers, you can actually increase the gamma motor neuron discharge, which will help us to get a better response for the reflexes. Is it clear? And one more thing, the root value is very important here, because by examining the reflexes, you will actually come to know the site of lesion. Fine. And any clinical examination, so that was about, we have completed the sensory system examination, the motor system examination, and the reflexes. Reflexes is again a part of motor system. So we have completed the sensory system examination and the motor system examination in the central nervous system. In the next week, I think probably we will be demonstrating how to examine for the cranial nerves, and that completes the entire examination of central nervous system. Okay. So please practice, as you, if you don't have the stethoscope with, means if you don't have the knee hammer with you, you please means you can take the help of your stethoscope and start practicing. Okay, and the position is very important. Don't hit the tendon. Okay, the stroke has to be delivered from the wrist joint, just like your percussion. We used to make you practice, isn't it? So just like your percussion, you have to deliver the stroke from the wrist joint. It is not from the elbow joint. If it is from the elbow joint, you will be hitting the tendon like this, hitting the tendon like this. So it has to be from the wrist joint, then only there will be smooth movement. And once you deliver the stroke, immediately it should be withdrawn. That means once you tap the tendon, immediately it should be withdrawn. And you have to observe for both contraction of the muscle plus the movement of the joint. Okay, contraction of the muscle also you have to observe, and also the movement of the joint. Okay, so any doubts you can start posting and we will take the questions. So that was about examination of reflexes. Since there are no questions, we will conclude the session here. Thank you.